Hello, बच्चों. We have started the discussion of paper 2025 NEET. Okay, I am Ambaresh Tiwari. I am going to discuss the zoology part of that paper. Okay, and now here is the Pallavi ma'am. She is going to take that chemistry part.
Slightly see कि how get the interaction with.
Hello, everyone. So here with last year was very easy. Now let's see.
Hello students, my name is Gaurav. I will be discussing of now we have seen fact that we are was a bit difficult, difficult or lengthy, moderate was not as it was last year easy, right? So many students they are just connecting with us, getting the entire discussion and how was the paper when we start the paper discussion.
Hi. So as Shivam Shah said, the paper was a bit on the difficult side. We are still having a look at the paper and hopefully get back to you on pretty soon.
First, we have Amisha ma'am at botany.
Hello students. That the paper was little bit tough. I know you have faced numerous problems. I got the feedback from the students, but nothing to worry. We are going to discuss the paper with you. We are going to see. Yes, I know the paper is little bit difficult, even in botany also. But I will say botany is a bit easy. We have already discussed all the things that have already come in your this exam. I know that there's a pedigree question. You won't be able to solve at that pressurized moment. Let's discuss over here and don't worry, everything will be fine. So now we are going to discuss. We are going to start with the botany and zoology discussion. Let's start with the first question.
But before starting the discussion, let's talk about the paper of zoology. There are some questions not directly 30. Here we by watching the, I think that there are not even that can 720 out of 720. Okay, so there are few questions which are very out of NCERT. So whatever you have done, don't take any tension. Let's see one by one. We are going to discuss the questions, and then after the discussion, you can find out that what the mark you get in the exam. And don't take any tension about that. Whatever the cutoff is, cutoff will down this year. That is 100 and sure. Okay, so let's start with the discussion of the first question from the biology section. That is the 91 question. Let's open it.
So your first question is from botany. The question says that the complex 2 of mitochondria electron transport chain is also known as. See, we know what is complex 2. Complex 2 is an enzyme that also participates in Krebs cycle. Yeah, right? This is a common enzyme which is a part of ETS plus it is a part of Krebs cycle. So your answer is going to be succinate dehydrogenase. This one is very easy. So moving to the next question.
Yeah, this is the question from that biotechnology part. Here they are talking about the PCR polymerase chain reaction. So here there are the few options given that have some equations like polymerase chain reaction PCR amplifies DNA following the equation. Which equation should it follow? It is the second 2 to the power n. As in the first generation, that that DNA fragment is used, that forms 2 DNA, and as per 2 it can form 4 forms 8, and that is how this is the correct answer, second answer. Now, what are the potential drawback in adoption of the IVF method? See, they are talking about the drawbacks of adoption. They are talking about the drawbacks of adoption. See, focus on this point. Okay, the first is what? The high fidelity risk to the mother. See, it cannot be possible because during the process of IVF, the mother remains in under observation of the doctors. Okay, so this cannot be the correct option. Okay, next one is the expensive instruments and reagents. Yes, it is very expensive. It is already written in your NCRT. Okay, here the husband and wife necessary for being donor. Okay, it should not be again the correct option, because why? In the condition, if the husband or wife is not able to produce the ovum, or they cannot provide the environmental condition for the development of embryo, in that condition we choose that IVF. That's why this cannot be the correct option. Okay, so let's move on the next one, that the less adoption of orphans. Okay, that is the correct option. Okay, so if the person is using IVF, in that condition, if the couple do not have that progeny, they can take the orphans, they can adopt the orphans. But if they are using the IVF, they will not go for opt for this adoption. Okay, the next one is not available in India. This is wrong. In India it is available. So, and F, the next one is the possibility that the early embryo does not survive. Yeah, this is the condition. This is the condition. Okay, so there is a possibility in the early embryonic stage. In the early embryonic stage, what happens, that the miscarriage happens. Okay, so this is the drawbacks, that B, D, and F. So the option will be the one only. Okay, so let's move on the next question. What is the name of the blood vessel that carries deoxygenated blood from the body to the heart in a frog? In a frog. That is the question. What is the name of the blood vessel that carries deoxygenated blood from the body to the heart of the frog? So already in frog, yes, fourth option, it is the correct. So it is the vena cava. Vena cava carries the deoxygenated to the right auricle of the heart of frog. Okay, that's the correct option. Okay, next move on the next question. Which of the following statement refers to reductional biology? See, in reductional biology, what happens? We break down the cells in chemical constituents, and then we study the relationship of that chemical constituents, how they react with each other. Okay, so the first option is giving here the physicochemical approach to study and understand the living organism. No, it is wrong. We do not study the physiological processes. Okay, here thus it is physicochemical. Physiological approach to study the understanding living organism. The chemical approach is to study. Chemical approach is, yes, it can be the option. So in option 1 and 3 we are confused a bit more. So the first option is more probably the correct answer. Okay, okay, it is given in the NCRT summary of the cell biology. It is a physicochemical approach to study and understanding living organism. Okay, so moving on the next question. So it is given that in RNA world, so it is from botany. Okay, so ma'am will.
Okay. So now this is the botany question. Okay. For botany's discussion, one sir will also be there with me. Okay. Udit sir. Sir, please join me. Okay. So this is the botany. All it's the botany question. Okay. So let's discuss it.
So in the RNA world, RNA is considered as first genetic material evolved to carry out essential life processes. RNA acts as a genetic material and also as a catalyst for some important biochemical reaction in the living system. That is correct. Being reactive, RNA is unstable. Yes, as we know that on the second position, one extra OH group is present. So this is right. Being reactive, RNA is unstable. Second point, DNA evolved from the RNA and is more stable genetic material, is double helical strand being complementary. Yes, resist changes by evolving. pairing mechanism. In the light of the above statement, choose the most appropriate answer from the option. So as we have seen the statement 1 and statement 2. So statement 1 says the RNA world, RNA is a considered first genetic material. Yes, we can say, and evolved to carry out essential life processes. So we have seen that so many life processes are, it also behaves as a catalyst, it also behave as an enzyme. So we can say it's biochemical processes evolved around RNA. And DNA, we already know that DNA is more stable than RNA. Now let's see what are the options. Both statement 1 and statement 2 are correct. Let's see. Both statement 1 and statement 2 are incorrect. And statement 1 is correct, but statement 2 is incorrect. And statement 1 is incorrect, but the statement 2 is correct. So what I think is statement 1 is correct because it says that RNA is the first genetic material and it is unstable. And second statement is also correct because it says DNA is more stable, and because of its complementary nature, and resist changes by evolving repairing mechanism in the light. So both the statement, what I think is correct.
See students, in the class also we have discussed this RNA world, and I have told you that because during the origin of life there was a reducing environment, and in reducing environment we know that RNA is much more stable as compared to DNA. Okay? Plus I have told you why it is considered RNA as a genetic material in the previous time. Okay? Because of its specific properties like catalytic. In catalytic, we have already discussed that it is going to be going to participate in the translation mechanism, because RNA is going to act as enzyme, and that enzyme is known as peptidal transferase. We have already discussed in the class. And the second in the splicing, which is the most important function, and it helps us to study the evolution of RNA, and also we know that DNA has been evolved from RNA. So both the statements are going to be the correct. So its option is first. So move to the next question. Now epiphytes. Epiphytes we know. See, there is a difference in epiphytes and parasite. Parasites are going to suck the material from the main plant, but epiphytes, they just require help. Epiphytes, example orchids, as we remember. Epiphytes that are growing on a mango branch is an example of which of the following? So epiphyte is taking just the help. It is not consuming any material from the mango plant, mango branch. So the answer is going to be commensalism. Is it correct, sir?
Because mango is not harmed and it's not getting any benefits from epiphytes, and epiphytes are not harming mango, so that answer is commensalism. Mutualism we cannot write because both should get some benefit from each other. Okay? Predation, of course not.
Statement. Okay. First is the eukaryotic ribosomes are ATS. Yes, of course we know that eukaryotic ribosomes are ATS.
Yes.
And prokaryotic ribosomes are 70S. This is correct. Yes. Then this is correct. Then each ribosome has
two subunits. That is also correct.
And smaller subunit. So this is B also correct. Then two subunits of 80S is 60S and 40S. This is also correct. While that of 70S are 50S and 30S. So C is also correct. So A, B and C are correct. Okay. Now, rather than studying all the statements, we can also save our time in this question. Just we can see they have given 3 or more than 3. So they have given 3. Okay. So just we can solve by looking at the options A, B and C. So it is going to be the first option.
And we can clearly see D option cannot be correct because 80S, 60S and 20S is wrong, and 70S, 50S and 20S wrong. E option, two subunits of 80S are 60 and 30S. This is also wrong. Okay? Our answer is first. Let's move to next question. Which of the following is example of ex situ conservation? So ex situ conservation, we can see we can say it's not a on-site conservation. We are conserving the threatened species in a proper different area. So national park, wildlife sanctuary, zoos and botanical garden protected areas. So what I think is XC2 conservation, national park cannot be the right answer. Wildlife sanctuary no. Zoos and botanical gardens, it's a conservation. Now let's move to next question. The primary source of energy in an ecosystem is solar energy. Yes, we can say it's the solar energy which is the primary source of energy. The rate of production of organic matter during photosynthesis in an ecosystem is called as net primary productivity. So rate of production, and this statement for short, and statement the rate of production during photosynthesis is a ecosystem is net primary. Let's see. For me, statement 1 is correct and statement 2, ma'am, is also correct. Yes. So it's called as gross.
Yeah.
The statement 1 and 2 are correct. Both the statement 1 and 2 are incorrect. Statement 2, final answer. Third. Hmm. 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 Primary productivity. Yes. Yes. Okay. Okay. Actually, are not very confused and do like a reward for the organism or the bees. Right? Of pollinated flock. Because the thing is, wind and water do not have a particular direction. This honeybee have direction. Honeybee knows that I want to sit on this. I will next. What? That nation. Average.
Natural, right? The that if the concentration goes above 30%, the yeast is going to die by itself. So,
Itself.
poisoning itself. So, if we talk about whiskey, if we talk about brandy, if we talk about rum, they are distilled. Only beer is going to be the non-distilled. Clear? So, the answer is going to be third.
In this option 1,
More than 13.
it won't be possible for yeast to keep producing the alcohol more than 13. And nature of the flower, G stands for the inferior ovary. As we can see, right, inferior ovary, this is
See, for this is
actinomorphic.
actinomorphic, we all know, and the six notes zygote. To be superior ovary. This is the sign of superior. The dash is above G, it is the sign of inferior ovary. We are going to solve.
No,
What is going
incorrect.
correct?
Hmm?
With G and below Z. To be the first statement is incorrect and the second statement is correct. Statement 1 is incorrect and statement 2 is correct. So fourth option, it is going to be option. Now moving to the next point, streptokine is produced by the microbes chapter. Microbes. Now the answer it is doesn't related to the liver disease disease treatment. So the answer is going to be the fourth one, removing clots from the blood vessels. So for 105, the option is going to be the fourth one. Now moving to the next question. Which chromosome in the human genome has the highest number of genes? See, this is very easy question. We know it is chromosome number 1. So chromosome number 1 has the highest number of gene. I suppose 2986 if I am not wrong. Okay, so the answer is going to be the third option. So now moving to the next question. This is from zoology. Now next, which one of the following phytohormones promote nutrient mobilization? Nutrient mobilization which helps in the delay of leaf senescence in plants. It is related to which chapter? Plant growth and development. And the answer is going to be cytokinin. The answer is going to be cytokinin. See, in your NCERT in plant growth and development chapter, okay, when you are going to study cytokinin, the last statement they have given that cytokinin is involved in mobile nutrient mobilization and it is going to delay leaf senescence. Now moving to the second question. This is also, I think, zoology. Then this is also a zoology question. Now next is, given below are the stages in the life cycle of pteridophytes. Arrange the following stages in the correct sequence. Now see, in the class we have already made one life cycle. Yes or no? Let's draw the life cycle again. First of all, pteridophytes are sporophytes. Pteridophytes are going to be sporophytes. And these sporophytes are going to possess sporangium. In sporangium, there will be spore mother cells. These spore mother cells are going to perform meiosis. After meiosis, they are going to form spores. Now, spores are haploid. Spores are haploid. Spore mother cell is diploid. Now, this haploid is going to form a gametophytic stage which is known as prothallus in pteridophytes. Now, this prothallus is going to form archegonium and antheridium. We know antheridium is going to form antherozoids, male gametes, and archegonium, egg cell, syngamy, embryo, and zygote, and then a complete pteridophytic plant. Now see, now we are going to see the statements. Tick. Prothallus stage, it is a gametophytic stage. It is going to be at the other way. First step, first we are going to see which is going to be the first step. First step is meiosis in spore mother cells. See, spore mother cell, it is going to perform meiosis, and then spores are going to be formed. That spores are going to develop into prothallus. So first stage is going to be B. Then fertilization, fertilization is afterwards. Then formation of archegonium and antheridium in gametophyte. Yes, transfer of antherozoids and antherozoids to the archegonium in the presence of water. So in these two statements, first is going to be the formation. The second one is going to be the formation. Then the transfer of antherozoids to archegonium in the presence of water. This is going to be the third option. After that, fourth is going to be, sorry. I didn't saw prothallus stage. Got confused. Sorry. So first, this is going to be the first one. Then prothallus. This is going to be the second one. Then there is a formation of, this is third, this is fourth, and then this is fifth. So B, first is going to be B, then the second one is going to be A, so it is BA, here also BA, then the formation of archegonium, then it is going to be D, so it is D, then the transfer, and then there will be fertilization. So the answer is going to be first. This is zoology. Then which of the following organism cannot fix nitrogen? Let's move to the nitrogen fixation. See, this is not out of NCERT. It is microbes chapter. It is microbes chapter question. Anabina Nostock, nitrogen fixing. Tick. Then Oscillatoria, nitrogen fixing. Check Azotobacter, nitrogen fixing. Volvox, Volvox is, no, it is a colonial forming bacteria. So its answer is going to be D, second option. Volvox, actually this is from zoology. Okay, but the first option is incorrect, that we can say transfer RNA and ribosomal RNA do not interact with mRNA. This is incorrect. In translation, we see that ribosomal RNA is also interacting with the mRNA and transfer RNA is also interacting with the mRNA, because mRNA is going to have codons and tRNA going to have anticodons. Though this is zoology question. Again, this is zoology question. Now, I think this is also zoology question. This is also your enzyme portion, zoology. Now, find the statement that is not correct with regard to the structure of the monocot stem. Now, we have to say which is not related to monocot stem. We know this is the transverse section. Outermost is going to be the epidermis. Outermost is epidermis and inner to epidermis, we have hypodermis. Now, epidermis is basically made up of parenchymatous cell, but in the case of monocot stem, this hypodermis is going to be made up of sclerenchyma. We know that it is going to be made up of sclerenchyma. Then we have scattering of vascular bundles. Vascular bundles are scattered and the complete area is covered by parenchymatous cortex. Now see, hypodermis is parenchymatous. The first statement, we got it, that hypodermis is going to be sclerenchymatous, not the parenchymatous. So over here, we got the first, our answer, it is first. So we don't have to read all the statements. Why to waste our time in reading all the statement? But we know vascular bundles are scattered in the case of monocot stem. Vascular bundles are conjoined and closed. Why closed? Because they are not going to perform secondary growth. No secondary growth is seen. And yes, phloem parenchyma is absent in the case of monocots. So the most appropriate answer is going to be the first. Now moving to the next question, the correct sequence of events in the life cycle of bryophytes. Now bryophytes are, first of all, gametophyte. Bryophytes are gametophyte. Now these gametophytes are going to produce antheridium and archegonium. Antheridium is going to produce antherozoids. Archegonium is going to produce egg cell fertilization with the help of water. Zygote is going to form and then embryo. Now bryophytes are considered as the first embryophyta. Why? Because first embryo is going to be produced in bryophytes. Now this bryophyte is going to, embryo is going to produce sporophyte. This sporophyte is dependent on gametophyte. Now the sporophyte possesses capsule. This is the structure. We know that this is the structure and this is going to be the capsule. Now inside capsule there is spore, mother cells. Spore mother cells are going to perform meiosis, then spores, and then protonema, and from protonema then you will get the bryophytes. Now this protonema, we know this is seen only in the case of mosses. It is absent in the case of liverworts. Now let's see. Fusion of antherozoids with egg cell. Yes. Attachment of gametophyte with the substratum. Then we have marked this. Okay. Then reduction division to produce haploid spores. This is going to be the at the last. Formation of sporophyte, then release of antherozoids in the water. Now first of all, it is going to be the attachment of gametophyte to substratum. This is first. Now moving to the second, release of antherozoids into water. This is second. After that there will be fusion. This is going to be the third stage. Then after third, then we have formation of sporophyte, fourth, and the last is going to be the C, reduction division to produce haploid spores. So first B, these two are there. Okay, so we have to select from them. Then after B it should be E. After E it should be A. After A it should be, fourth one it should be D. So over here third option is going to be the correct one. C is the last one. So over here it is third option. Now next, I think this is related to zoology. Then yes, now moving to another question of botany. Centromere. Moving to the centromere. Now centromere is going to participate in cell division. Cilium, Cilium, it is cell movement. Then cristae, it is a part of mitochondria, and cell membrane, it is made up of phospholipid bilayer. So the answer is going to be for A, it is second. Let's see, for A it is second. So this and this cannot be there. We have a tie between second and third. So first is A, second option. Then moving to the B, they are telling cell movement third. So it is going to be the third. So fourth option. So according to me, it is going to be the fourth option. In just second, we got the answer. For C, it is first. We can recheck. For the D, it is fourth. So for 121, it is fourth option. Now moving to another question. Yes, now this is very complicated because you have to learn. Okay, now moving to, first of all, we are going to see chlorophyll A. Chlorophyll A is blue-green. Check, chlorophyll A is going to be blue-green. Let's see, A third. Now we have a tie between first and second option. Now moving to chlorophyll B. Now tell me, chlorophyll B is going to be yellow, green. So B is first. Xanthophylls, yellow color. Xanthophylls are yellow color, and carotenoids have yellow to yellow orange. So the most appropriate correct answer is going to be the second one. So for 122, it is second option. Now this is again, it is zoology. Now moving to the botany question. 124 is botany. In the seeds of cereals, we are talking about cereals like maize. The example, the most common example is maize. The outer covering of endosperm separates the embryo by a protein-rich layer. This is known as, we know, aileron layer. Tick. So the correct answer is going to be aileron layer. Next question. I think this is again zoology. This is also zoology. We have zoology teachers also, so I am asking them, okay, whether it is zoology or not. Now, this is botany question. So, we have again assertion and reason. A typical unfertilized angiosperm embryo sac at maturity is 8 nucleated and 7 celled. Yes, this is central cell. Then we have 3 cells towards chalaza end, that is antipodal cells. Then at the center we have polar nuclei. There are 2 polar nuclei. Then towards the micropilar end we have synergids, and this is X cell. So yes, it is 8 nucleated and 7 celled. It is 8 nucleated and 7 celled. The first option is going to be the correct, yes or no? Now, the egg apparatus has 2 polar nuclei. Egg apparatus is made up of. This complete structure is termed as egg apparatus. So, do you find polar nuclei in egg apparatus? I don't think so. So, this is going to be false. So, assertion is correct, but the reason is false. Let's see where it is given. Third option. Assertion is true, but the reason is false. Now, moving to the another question. A specialized membranous structure in prokaryotic cell. This is, they are telling you specialized membranous structure. Whenever we hear, okay, listen to this prokaryotic cell and specialized membranous structure, we strike with the 2 structures. One is chromatophore and the other one is going to be mesosomes. Now, we are moving forward with the question. Let's see, we have to decide between 2, whether it is chromatophore or mesosomes. Now, which help in cell wall formation, DNA replication and respiration. See, this is the best to find out the answer. Respiration, from this we say that it is analogous to mitochondria. So your answer, the structure which is analogous to mitochondria in prokaryotic cell, your answer is going to be mesosomes. So the correct option is mesosomes. Now, moving to the next question. This is eukaryotic transcription, post-transcriptional mechanism. Which of the following are the post-transcriptional events in an eukaryotic cell? Transport of pre-mRNA to the cytoplasm prior to the splicing. Correct, incorrect? This is incorrect. Okay, first the hRNA is converted to, hnRNA is converted to mRNA, and we know the processes. First is going to be the capping, then splicing, and then it is tailing. Let's find out the correct answer. Removal introns and joining of exons? Yes. Addition of methyl group F5' end of the hnRNA? Tell me, this is correct or incorrect? This is correct. Capping is done at the 5' end and tailing is done at the 3' end. Now, addition of adenine residues at 3' end of hnRNA? This is correct. Then base pairing of two complementary RNAs? No. This is not a part of post-transcriptional mechanism. So, we get B, C and D. Let's find out the correct option B, C and D. Where it is written? Second option. The correct is going to be the second option. Moving to next question. What is the pattern of inheritance for polygenic trait? First of all, polygenic and inheritance do not follow Mendelian law. Do not follow Mendelian laws. We know that. Yes or no? Tick. Now moving to the statements. Mendelian inheritance pattern. Are you sure? No. Non-Mendelian inheritance pattern? Yes. Autosomal dominant pattern? No. It is a character. Polygenic inheritance. They are showing the characters, especially in humans. Most of the features, most of the characteristics of humans follow polygenic inheritance. So this cannot be the part. Then X-linked recessive? No. Neither Mendelian. So our option is going to be non-Mendelian inheritance pattern. Moving to the next question. This is not the part of botany. Then this is, yes, this is 5 kingdom classification system, biological classification. Each of the following characteristics represent a kingdom proposed by Whittaker. So we know that Whittaker has given 5 kingdom classification. First kingdom is Monera. The second kingdom is Protista. Then we have fungi. Then we have plantae. And then we have Animalia. Biological classification chapter, second page. We have a table there. Now we have to compare this with the statements given here. Multicellular heterotrophs with cell wall made up of chitin. They are talking about fungi. Fungi is going to be the third part. So I am writing over here A. Heterotrophs with tissue, organ system level. We know organ system level is present in kingdom Animalia. So B is going to be Animalia. Then prokaryotes. There is only one kingdom which is dealing with prokaryotes, and that is Monera. So C is going to be Monera. Eukaryotic autotrophs with tissue, organ level of body organization. We know that plants do not have tissue system, organ system. So D is going to be the part of plantae. The statement is related to plantae. Then eukaryotes with cellular body organization. This is Protista, E. So our sequence is CE, ADB. So let's find out CE, ADB. It is second option. So over here, this is going to be the second option. Now, who is known as the father of ecology in India? See, though we have not studied in class, I know, okay, but it is given in NCERT when the introduction is of ecology chapter starts there. So its answer is going to be Ramdev Mishra. List 1 and list 2. Now we have to match the following. Hershey and Chase, they gave the unequivocal experiment to prove that DNA was a genetic material. So let's find out where they have given genetic material. So for A it is 4th. Then euchromatin, lightly and loosely, lightly, you can say lightly stained and loosely packed. So euchromatin. Third option. Then Frederick Griffith. Griffith has performed the experiment in mice and Streptococcus pneumoniae. Heterochromatin, densely packed and darkly stained. Let's find out the correct option. For A, it is 4th. We have the tie between 2 and 3rd. Then euchromatin, it should be 3rd option. For B, it is 3rd. So our most appropriate is going to be the 3rd option. A with 4th, B with the 3rd, C with 1st, and D with again 2nd. So the answer is going to be the 3rd option. Now moving to the next question. I think this is zoology. Then this is also zoology. This is botany. Though it is a part of biomolecule, but we can also solve this question, because we study molecular basis of inheritance. Adenosine, this is nucleoside. We know that this term is used for nucleoside. Adenylic acid, phosphate is attached, that is why it is acid. So this is going to be nucleotide. Adenine, this is nitrogenous base, and alanine, you know, it is amino acid. Now let's find out the correct option. Adenosine is going to be nucleoside, 3rd. Adenylic acid, nucleotide. Adenine, nitrogenous base, and alanine, amino acid. So for A, it is 3rd option. Again, we have tie up between 1, 2nd, and 3rd. So adenosine, 3rd. Adenylic acid, 2nd. Again, now we have a tie up between 2 and 3rd. Adenine, it's nitrogenous base. Now we can see which is going to be C. It's going to be the 1st. So our correct option is going to be 3rd. Now the correct option is 3rd. And D is 4th. Now this is also, I think, zoology question. All living members, this is also zoology question. Let's talk about this statement. Given below are two statements. One is labeled as assertion and the other one is labeled as reason. The primary function of Golgi apparatus is to package the materials made by endoplasmic reticulum and deliver it to the intracellular targets outside the cell. Yes, this is correct. This is going to be the correct. Tick. Vesicles containing material made by endoplasmic reticulum fuse with the cis phase of the Golgi apparatus and they are modified and released from the trans phase of the Golgi apparatus. This is also correct. Now the main question is, both are correct, we know, but we have a tie between first and second option. So this is correctly explaining why vesicles are formed, because they have to modify and then Golgi is going to take it, distribute where these vesicles are required. We know that Golgi apparatus is going to act as a traffic police of the cell, right? So according to me, the answer is going to be both A and B are true and R is going to be the correct explanation of the A. So it is 140, it is going to be the first option. Now match the following again. Scutellum. Scutellum is present in monocot seed. We know that it is present in monocot seed, papery like structure. Non-albuminous seeds, Fabaceae family. ठीक है? Epiblast, remains of secondary cotyledon. Perisperm, seeds which possess the persistent nucellus. Now see which is matching scutellum. It is going to be rudimentary cotyledon or cotyledon of monocot seed. It is going to be cotyledon of monocot seed. Non-albuminous seeds, Fabaceae family, groundnut. Epiblast, rudimentary, remains of secondary cotyledon. So the option is going to be rudimentary. Okay. Then perisperm, perisperm means persistent nucellus. Now for A it is second. For A it is second. Again we have a tie between first and fourth. Now moving to the B, non-albuminous C, it's going to be groundnut. A is second and then B is going to be third. So our correct option is going to be, no, again we have this second and third option. Then we are going to find out our correct option with the help of C and D. For epiblast it should be fourth. Scutellum, for scutellum it is second, first and fourth. Then for B, non-albuminous seeds, it is groundnut, third. So it is first option. Then for C, it is rudimentary cotyledon, and for D, it is persistent nucellus. So the first option is going to be correct. I got confused with this second and third. So it is the first option. Now again, this is zoology question. Then this is also zoology question. Zoology. Now let's do the question of genetics. It's simple. Okay. They are telling you it is independent assortment and the ratio of independent assortment is 9:3:3:1. So let us find out where is 9:3:3:1 phenotypic ratio, and it is third option. So the answer is going to be 9:3:3:1. Check. See, simple. It is simple. Capital R, capital R, capital Y, small r, small r, small y, small y. Over here, okay, when we are going to do cross pollination, we will get hybrid capital R, capital Y, small y. Tick. Then we perform selfing, and after selfing, we are going to get phenotypically four types of progeny. Okay. This is round, and this is, they have given round and yellow. Sorry. Round and yellow. Then round and green. Then we have wrinkled and yellow. And then we have wrinkled and green. Wrinkled and green. So their number is 9. This is 3, 3, and this is 1. So the correct option is going to be phenotypic 9:3:3:1. Histone proteins. This we have discussed numerous times in class. These histone proteins are positively charged proteins. Positively charged proteins. And because they are positively charged proteins, so they should be made up of positively charged amino acids. That is lysine and arginine. So the first option is the correct option. This is zoology. Again, this is zoology. Again, this is zoology. I think zoology they have asked many times. Tick. Now, which of the following is the unit of the productivity? I don't think I have to explain anything in this. It is going to be the fourth option. Tick. This is the unit to determine the productivity. Okay, primary productivity. This is again in ecosystem chapter. Now, this is zoology. Now, this is, I think, okay, like, see, I know that you won't be able to solve it, this question, okay. This was not given in your NCERT, it is out of NCERT, okay, and even in the class we have not discussed any question like this because we have seen that they were asking simpler questions. Though it is simple, but over here there is some different mechanism that they have followed. What different mechanism? See, they have given you the carrier, they have given you the deceased, everything they have given you in this pedigree. Okay, so you don't have to find out which is going to be the carrier, which is going to be the deceased. Now the trick what they have played, they rather than asking you or, they have asked you about and. When we solve for or, it is multiplication, and when they give you and, this is addition. So they have asked you for both. Let's see the question and then I'm going to explain you. With the help of the given pedigree, find out the probability for the birth of the child having no disease and being a carrier. Rather than or, they have given you and. Or is a multiplication and is a addition. Okay, this is not in your NCRT. They have not given anything like that. Okay, but they have given the question. We have to solve the question. Okay, just remember this point. It will help you in your further studies also. When or is given, it should be multiplication, and is given, it should be addition. Now directly we can solve. They have asked you the last one in F3 generation. Okay, in F3 generation they have given you the cross. This is carrier XXC crossed with XY, and this X is deceased. So this is male is completely deceased and the female is carrier. So when we are doing a cross, we are getting XXC, a carrier female, then XY, non-deceased male, then XCXC, deceased female, and XCXY, this is deceased male. Now they have asked you and no disease and being carrier. So no disease condition is this non-deceased condition, and this is carrier. Okay. Now it is going to be 1/2 plus 1/2. It is going to be 1/4. So according to me, the answer is going to be 1/4. Okay, let me drink some amount of water, then I'm getting back to you. Just give me 2 minutes. Okay, so let's start it again. Okay, this concept was correct, but it was my mistake that one thing I just left it out. I took male and female, though they have not given anything male over here. They have just asked you the progenies. And when we are talking about the progenies, each progeny has the probability of 1 by 4. So let's solve it again. Okay. We are going to follow the same rule, or when it is given or, then it is multiplication. When it is given and, it is addition. Now, when we are solving for this, okay, it is x carrier, xcy, this is deceased. Now, this is xc, this is xc and y. Now, this is xc, this is xy, this is xc, and this is xcy. Now, in the question, they are not telling you that whether it is a male or a female progeny. When they are telling you male and female progeny, then we can take 1 by 2. But now, we have to collectively solve this. According to the progeny, the carrier is 1 by 4. The non-deceased condition is 1 by 4. And the deceased condition is, this two are deceased among the 4. So the deceased condition is this. Now they are telling us that we have to find out one no deceased and the other being the carrier. So the other is carrier and the other one is going to be the deceased. It is 2 to the power 2 by 4, sorry. It is 2 by 4 and the answer is 1 by 2. So the answer is going to be 1 by 2 rather than 1 by 4. So this is the whole detailing. We don't discuss this thing in the class, neither it was asked before. Okay. This is something new question that we are discussing. Now the next one, again, assertion and reason. Now assertion, cells of tapetum possess dense cytoplasm and generally have more than one nucleus. This is correct. This is true. Presence of more than one nucleus in the tapetum increases the efficiency of the nourishing the developing microspore mother cells. This is true. That is why the tapetum is going to have more than one nucleus. So this assertion is correct, reason is correct, and the reason is the correct explanation of the assertion. So our answer is going to be first. Both assertion and reason are true and reason is the correct explanation of assertion. Now moving to the next question. How many meiotic and mitotic divisions need to occur to develop for the development of a mature female gametophyte from the megaspore mother cell in an angiospermic plant? They are asking you to form a embryo sac from megaspore mother cell. We know that there will be 1 meiosis and there will be 3 mitosis. Then only this complete embryo sac structure is going to be formed. Let's see where this option is given. This is third option. 1 meiosis and 3 mitosis. Now, moving to the next question. Which of the following is the example of zygomorphic? Most common Fabaceae family members. P. Answer is going to be third. Even Poaceae members are also zygomorphic. Again, this is zoology question. Okay. Now, moving to 157 question. See, we have talked this pig and wasp mutual relationship in the class. Okay. In sexual reproduction of flowering plant, and we have seen that the flower is dependent upon the wasp for the pollination, and the wasp is getting a protective covering, or you can say they are getting protection for their larva, plus they are getting the food also from the plant. So let's see, okay, what they are telling us. Fig fruit is a non-vegetarian fruit? No, it is a vegetarian fruit. Okay, as it has enclosed a fig wasp in it. No, it doesn't matter. The fruit is always going to be vegetarian. Now, fig wasp and fig tree exhibit mutual relationship. This is correct. As fig wasp complete its life cycle in the fig fruit, and fig fruit gets pollinated by the fig wasp. Now, life cycle. If we talk about the life cycle, yeah, both the statements, but the fig first is incorrect. Yes, first is statement is incorrect, and the second statement is yes, correct. Completes its life cycle in fig fruit. Yes, okay. So it is required for the development because of the food. So this is going to be the correct. So the statement 1 is incorrect and the statement is correct. So I think statement 1 is incorrect and the statement 2 is correct. Fourth option. Now moving to the next question. What is the main function of spindle fibers? Spindle fibers are required for the separation of the chromatids in the chromosomes. So over here the answer is going to be the first option. Now next, which of the following is the characteristic feature of gymnosperms? Gymnosperms, they do not have ovary. No ovary is there. Seeds are naked. So our answer is going to be second, seeds are naked. Now next, this is zoology question. This is again zoology question. This is botany. Match the following. Now, this is salvia and this is salvinia. Okay? There is a difference in salvia and salvinia. Salvia is angiosperm and salvinia is pteridophyte. So for pteridophyte, it is fourth. A ka hai fourth. Sorry, for A it is fourth. For biophytes, it is polytrichum. For angiosperm, it is salvia. For gymnosperm, it is ginkgo. Now let's match it. For A, it is fourth. We have a tie-up between 2 and 4. For bryophytes, it is going to be the third option. For angiosperms, it is going to be the first option. So A fourth, B third, and C is first. Then D is second. So we have the second option over here. The option is going to be second. Next, who proposed that the genetic for amino acid should be made up of 3 nucleotides? This was given by George Gamow. The first experiments were from the first experiments the genetic code was being deciphered. So it was from the starting was done by George Gamow. Moving to the next question now. Next question is again match the following. Evil quartet. They are related with the loss of biodiversity. For A, it is going to be the third. Cause of biodiversity loss. A is third. All the fourth options we have A as a third. Now XC2 conservation. XC2 conservation is karyopreservation. For B, it is first option. Second option, we got it second option. For C, lantana, we have alien species. This is alien species. And dodo, it is extinct species. So the most appropriate answer is going to be the second option. Next, this is zoology question. Again zoology question. Again this is zoology question. In bryophytes, Gema is asexual bird. Gema is asexual bird. Okay. So our option again it is going to be asexual reproduction. Direct question. This is I think zoology. Now let's move to 170 question botany. In ecosystem, there is unidirectional flow of energy from sun. Energy of sun from producers to consumer. Correct, incorrect? This is true. So statement 1 is true. Now, ecosystem are exempted from second law of thermodynamics. No, ecosystem follows second law of thermodynamics. This is false. So statement 1 is correct, but statement second is incorrect. Third option. So 170 we have third option. Next, which of the following statement about Rubisco is true? So Rubisco is involved in dark reaction and it is going to catalyze the carboxylation step. Plus, it has both the activities. Carboxylase acts as well as oxygenase activity. Let's see. It is active only in dark? No. It has the higher affinity for oxygen and carbon dioxide. Both higher affinity for carbon dioxide. Okay? When both oxygen and carbon dioxide are present at the same concentration. Is of water? No, it is oxygen. If is going to catalyze the carboxylation of ribulose 1,5 bisphosphate. No, this is zoology. Oh, plant growth regulators as well as is the request for different before redifferentiation there should be this is incorrect. Apical dominance the growth of lateral bud. No. If there is apical dominance, lateral buds are not going to get formed. So this is also incorrect. So we have three statements correct, A, B and C. So our most appropriate answer is going to be the first one, A, B and C. Now moving to the next question. Which factor terminate required for initiation? This is also zoology. Formed. ठीक है next क्या
There is issue with that. Okay. Now it is good. No, that pin was had came out from that. Yes. Hello. Okay. So probably there was no sound in the video. I am very sorry for that. Okay. Let's again start with that first question. Okay. We have already did. Okay. So let's start it again. Let's start it again. There was issue in that plug. Okay. So so this is the question from yeah, this is the question from the biotechnology part. Okay, so this is the question from the biotechnology, the PCR. In the PCR, what we do, we amplify the DNA. Okay, so from one DNA fragment we break it into two templates, and in the next stage what happens, we add a primer on it, and in the last step, in the chain extension part, what happens, the two new daughter DNA are formed. So if we see here that one DNA forms two DNA, if the same cycle is repeated multiple times, so we can get that 2 to the power n number of DNAs, 2 to the power n number of DNAs in the process. Okay, if n is the number of cycles here. Okay, so this is the answer, correct answer, 2 to the power n. Now let's move to the next question. What are the potential drawbacks in the adoption of the IVF? So we have already seen that in high fatality risk to the mother, because the patient is, or the mother is under the observation of doctor. This is not the correct option. Okay, expensive, yes, the instruments and the reagents are very expensive in India and all the other countries also. Okay, so this is the correct option. Husband and wife necessary for being donor. Okay, so this is not the criteria, because in the condition when the husband or the wife is not able to produce the gametes, or if they are not able to provide suitable environment for the process of fertilization and also for the implantation, in that case they go for that IVF. Okay, so this is not the correct option. Again, the less adoption of orphans, yes, obviously if the couple is not able to produce progeny and if they are opting for IVF, in that scenario what will happen, that if they are opting for IVF, they will not adopt the orphans. Okay, so this is the correct option. Okay, not available in India, this is wrong. In India it is available. We are now towards the developing countries. Okay, so here next option is possibly that the early embryo does not survive. Okay, there is a possibility in the process of implantation sometimes, or even in early stages of pregnancy also, what we see that the miscarriages happens. Okay, so that is why the mother is needed to be in observation of the doctors. Okay, so here the correct option is B, D and F. So here this first option is correct. Okay, next question, let's talk about that frog. It is from the anatomy of frog. Okay, what is the name of the blood vessel that carries deoxygenated from the body to the heart in the frog? Okay, so if we talk about the deoxygenated blood, which is carrying the deoxygenated blood to the heart in the right auricle, is the vena cava. In humans also it is the superior vena cava and the inferior vena cava. Similarly in the frog also it will be the vena cava. Okay, so now let's move on the next question of zoology. This was from okay, here let's see. See, in this question there is a list of disease and the list of symptoms. Okay, so in emphysema, what we see, emphysema is caused by the smoking. okay, over a smoker, they have that sort of disease in the lungs. Okay, what happens in that, the thickness of that respiratory epithelium increases, and just because of that, the total surface area for the process of gaseous exchange decreases. Okay, so the here we have the damaged alveolar wall and decreased respiratory surface. So emphysema will be connected with the second option. Angina pectoris is the cause due to the heart attack, and it is the severe acute chest pain. Okay, so here we can connect this angina pectoris with that second option. In glomerulonephritis, if there is a suffix like itis, that means inflammation, and here it is of glomerulo, that means the inflammation of glomerulus. Okay, so the glomerulus can be connected directly with that inflammation of glomerulus of kidney. Okay, now tetany is caused by the deficiency of calcium ion in the body. Okay, and in this condition the muscle spasm is found. Okay, so now here the tetany can be connected with the first one. So here A second and B, I think it is third, and glomerulonephritis C, it is fourth, and D, it is first. So fourth option is correct. Now moving to the next question. Okay, here. Which of the following statement is correct about location of the male frog copulatory pad? Okay, so the male, we have studied in the class that the first digit of the forelimb of the frog have that copulatory pad. Okay, by the help of this copulatory pad, they stick on the female's back of back. Okay, for long duration of time, and the female continuously starts laying egg, and the male sprays the sperm on those eggs for the process of fertilization, and the fertilization is external here in the frogs. Okay, so the first digit of the forelimb is the correct option. Let's move on the next question. Okay, so here is the question. While trying to find out the characteristic of newly formed animal, a researcher did the histology of adult animal and observed a cavity with presence of mesodermal tissue towards the body wall, but no mesodermal tissue was observed towards the alimentary canal. What could be the possible coelom of the animal? See, when we talk about that classification of coelom, first of all we see that whether the body cavity is present or not, and then if the mesoderm lines entirely that body cavity, only then it is called the true coelomate. But in this case the mesoderm is lining only, that mesoderm is only mesoderm tissue towards the body wall is present, but no mesoderm tissue was observed towards the alimentary canal. That's why, because it is not completely covering the body cavity, that's why it should be the pseudo coelom. Okay, so here the second option will be correct, pseudo coelomate. Okay, now next move on the next question. Okay, so here we have the structure of sperm. Okay, and the different parts of sperm is given here, and the function of that, the part it contains is given in the list too. Okay, so head, if we talk about head contains the nucleus. Okay, and the nucleus contains the genetic material, so A will be connected with the fourth, and middle piece have that mitochondria, that ribbon like mitochondria, which is responsible for formation of ATP, so it will be the energy, and the acrosome, that have the enzymes for the process of penetration. Okay, so the acrosome will be connected with that enzyme, and the tail, which is responsible for mobility, so with this tail will be connected with the sperm mobility, that is the second. So here the A is should be with the 4, and B with that third. Okay, and C acrosome is with the first one, so first option will be the correct option. Okay, here next move on, let's move on the next question. Okay, here this is the botany question. Okay, now the cardiac activity of heart. The cardiac activity of heart are regulated by, see, the cardiac activity in humans particularly, if we talk about it, is myogenic heart. Okay, so primarily it will be regulated by the nodal tissues of the heart, but the influence of other hormonal influences, the regulation of by hormones, and the neural regulation is also found. For the regulation of heart activity by nervous system, we have the cardiac rhythm center on the medulla. Cardiac rhythm center on the medulla. Okay, so a special neural center in the medulla oblongata, that is the cardiac rhythm center is present on it. Okay, and next if we talk about the hormonal regulation, there are some hormones among this. The one hormone which is called the adrenaline, which can accelerates the heart rate and can also enhance the strength of contraction. Okay, so the adrenal medullary hormone, that is the adrenaline, that is the catecholamine, is responsible for regulation of it. Okay, so A here the nodal tissue, so A, B and C will be the correct option. Fourth, here it is written as that adrenal cortical hormone. So adrenal cortical hormones do not directly influence the regulation of heart activity. They can only regulate the blood pressure of the heart. They can influencing the reabsorption of salt and water. So they do not directly influence the heart activity. So here the A, B, C will be the correct answer. So here first option will be correct, A, B and C only. Now move on the next question. Okay, here the transfer RNA and ribosomal RNA do not interact with mRNA. You must have studied in the process of translation that in the process of translation that ribosome, that transfer RNA transfers carries that amino acids with it. Okay, ribosomal RNA is present in the ribosome by which that mRNA binds. Okay, so here all these three RNAs interact together. So here it is written that transfer RNAs and ribosomal RNA do not interact. That is the wrong statement. In second statement, RNA interference, RNAi, takes place in all eukaryotic organisms as method of cellular defense. Okay, so as we have already seen in the class discussion that the RNA interference process is seen in the eukaryotic cells. Okay, so this is the correct statement. So statement 1 is incorrect and second is correct. Where is the option? Statement 1 is incorrect and statement 2 is correct. Fourth option will be the correct option here. Now let's move on the next question. Here this is the plasmid. Okay, in the above represented plasmid, an alien piece of DNA, here in the alien piece of DNA is inserted at EcoR first site. Here the EcoR first is the restriction endonuclease and it is present inside the gene that is called the beta galactosidase. Now as we all know that beta galactosidase is responsible for converting X-gal into blue color pigment. Okay, so blue color pigment, what will happen? Okay, so in the above represented plasmid, an alien piece of DNA is inserted at EcoR first site. Which of the following statement will be chosen to select the recombinant colony? Okay, recombinant colony. Let's see, if we insert that DNA, foreign DNA inside the beta galactosidase, that beta galactosidase will become silent. Okay, insertional inactivation will be found here. Okay, and because of that the blue color will not be produced in those bacteria or those host organisms which are recombinant actually. So let's see the options. Using ampicillin or tetracycline containing medium plate, no. Blue color colonies will be selected. The recombinants will not be blue because of insertional inactivation. Okay, white color colony will be selected. Okay, so third option will be the correct option. Now fourth, blue color colonies grow on ampicillin plate can be selected because here we are inserting the gene inside the foreign gene inside the beta galactosidase producing gene. That's why that is the Z gene actually. That is the Z gene. You must have studied in botany in that lac operon model. Z gene produces that beta galactosidase. Okay, so here the insertional inactivation, because of the insertional inactivation of beta that Z gene, the beta galactosidase will not be produced. Okay, and that is why the white color colony will be the recombinant only. Okay, now next one. Which of the following genetically engineered organism was used by Eli Lilly to prepare human insulin? See, it is again from a question from the biotechnology. That Eli Lilly company uses that bacteria, that is the E. coli. E. coli is the bacteria. Okay, so answer directly will be one, bacterium. It is the very easy question, direct question. Now let's move on the next question. Name the class of enzyme that usually catalyze the following reaction. See here in this reaction what happens
here where G is represented as a group other than the hydrogen. Okay, hydrogen is not being transferred from S to S dash, S has. Okay, so here from G is transferred from S to S has. Okay, so functional group transfer is happening here inside this chemical reaction. So what will be that enzyme? The class of enzyme will be transferase. Let's see the option. The third option will be the correct option. Now move to the next question. Okay, which are correct? Okay, let's see first that whether they want the correct or not. Choose the correct answer from the option given below. Okay, they need that correct statement. So first, compound tomography and magnetic resonance imaging, that is MRI. CT, compound tomography, and CT scan and MRI detect cancer of internal organs, that is correct answer. Okay, now the chemotherapeutic drugs are used to kill non-cancerous cells. We are not mad that we are going to kill that non-cancerous cells. Okay, so this is not the correct option. Okay, alpha interferon activate the cancer patient's immune system and helps in destroying the tumor cells. Yes, this alpha interferon, what does it do? What alpha interferon do? It enhances the activity of what? NK cell. NK cell, it is the immune modulator. It is the immune modulator. Okay, so this is the correct option. Now the NK cell kills the cancerous cell. Okay, the NK cells kills the cancerous cell. Okay, so now here C is the correct option. Chemotherapy drug are biological response modifier. No, it is not correct. Chemotherapy drugs are used to kill the cancerous cells. They kill that cancer cells. Okay, or either they reduce that replication rate of DNA. Okay, and that is how they reduce the number of cancerous cells in the body. Okay, now in the case of leukemia, blood cell count will decrease. No, it will increase. Okay, in the cancer the rate of division increases. Okay, then how can it be possible that number of leukocyte will decrease? Okay, so here the correct option will be 1, A, C and E only. Where is the option? A and C only. E is again incorrect, na? Oh, sorry, E is incorrect. So A and C is the correct option. Fourth. Okay, now in the next question, let's see where's the zoology part. Okay, this is the question I think from embryonic development. Okay, so find the correct statement. Okay, now in human pregnancy, the major organ systems are formed at the end of 12 week. Yes, in first trimester major organs are formed. Okay, and that is why the first trimester is very much prone to that it is prone for teratogens. Okay, it is prone for teratogens. So A is correct. In human pregnancy, the major organ systems are formed at the end of 8th week. Statement A is correct. Because of that, again the B will be incorrect. In human pregnancy, heart is formed after 1 month of gestation. After 1 month of gestation, this is the wrong statement. Before the completion of 1 month, the heart is formed. Okay, in human pregnancy, limbs and digits develop by the end of 2nd month. Yes, it is the correct option. It is written in the NCERT. It is written in the NCERT. In human pregnancy, the appearance of hair is usually observed at the end of usually observed in the 5th one. It is again the correct option. Okay, so which option will be correct? A and E only. D is not correct. Why? In human pregnancy, the limbs and digits develop by the end of 2nd month. It is correct. It is given in the NCERT. And if we talk about this one, see, the heart is formed in 1st month of gestation. So we can include this also. A, C, D and E. C. End of 2nd month. 8th week, that's why it is written. So this option will be correct. A, C, D and E. A, C, D and E. Third option will be correct. Now, let's move on the next question. Here, in this diagram, it is given that which one is correct. Okay, they are representing here the reabsorption process in proximal convoluted tubule, here written P, and from the distal convoluted tubule. At the same time, they have represented here the process of selective secretion. Okay, here in option number first, you can see that water is not being reabsorbed in proximal convoluted tubule, but water is what selectively excreted. So this will not be the correct option. Here in second option, we can see the water is being reabsorbed here and here also. Okay, the water is being reabsorbed here, and again in the proximal convoluted tubule, bicarbonate and sodium chloride, water is being reabsorbed. Sodium chloride, water and bicarbonate, again it is also reabsorbed in the distal convoluted tubule. Hydrogen ion and ammonia is selectively secreted in the proximal convoluted tubule, and in the NCERT it is given that potassium ion and hydrogen ion is selectively secreted in distal convoluted tubule. So the second option will be the correct option. Okay, you can directly match it from the diagram of NCERT, it is directly taken from there. Okay, so in this diagram, this is the diagram of bioreactor. Let's see what the question say. Identify the part of bioreactor which is used as the foam breaker from the given figure. Okay, so the foam will be accumulated on the top layer, so C will be the structure which will be used as the foam breaker. Okay, correct. Okay, so the option will be fourth directly. Now let's move on the next question. Okay, which one of the following enzyme contains heme as the prosthetic group? So when we classify the enzymes at the time of studying the enzymes in the biomolecule, we study about the prosthetic groups. So heme is the prosthetic group with catalase. This must be a memory based question. The fourth option will be correct. Okay, now let's move on the next question. Okay, this is the question from cancer. Okay, neoplastic characteristic of cell refers to. What is neoplastic transformation? The formation of cancerous cell because of any carcinogen from the normal cell to carcinogenic cell or the cancerous cell is the process called neoplastic transformation. So what happens in the neoplastic cell, the cancerous cell? The mass of proliferating cell, yes, obviously it will proliferate very fast. Rapid growth of the cell, yes, it will grow very fast. Invasive and damage the surrounding tissue, yes, the cancerous cells damage the surrounding tissue. They compete for nutrition because of the rapid rate of division. Okay, those confined at original location, no, they are not confined at the particular location. Okay, that's why the option A, B and C will be correct. So here the second option will be the correct option. Got it? Now let's move on the next question. Okay, this is the question again from the biotechnology gel electrophoresis. Let's see what the first statement says. The DNA fragment is extracted from the gel electrophoresis can be used in the construction of recombinant DNA. Yes, it is the correct option. Okay, by the process of by the use of gel electrophoresis, we separate the desired gene on the behalf of the size. Okay, the small size DNA move faster towards the anode and the large size DNA moves slower towards the anode. Okay, that's why let's talk about the second statement. A smaller size DNA fragment are observed near a node while another large fragment are found near the well in the gel, agarose gel. This is again the correct statement. So both the statements are correct. Where is the option? Let's see. Both statement 1 and 2 are correct. So option first will be the correct option. Now let's move on the next question. Okay, so this is the question from actually zoology. Probably ma'am have discussed that. Okay, so let's see this question number 138. Consider the following. What we have to find out? We have to find out that correct option. Okay, the reductive division of the human. The reductive division of the human female gametogenesis starts earlier than that of the male gametogenesis. Okay, In females, the gametogenesis process starts in intrauterine life. That's why this is the correct statement. This is the correct statement. Now, the gap between the first meiotic division and the second meiotic division is much shorter for females compared to for males compared to females. See, when we talk about the gametogenesis in female, in the process of oogenesis, there is the arrests we find at two different stages. In first diplotin stage of prophase first, and then the metaphase second stage. Okay, and that is why, because of this arrest, the space, the timing between the two successive divisions increases in female. So B is also the correct option. In males, the division continues. There is no any sort of arrest found here. Okay, now the first polar body is associated with the formation of primary oocyte. It is wrong. It is connected with the formation of secondary oocyte. Okay, first primary oocyte is just formed by the growth, after the growth of what? Oogonium. Okay, so this is the wrong statement. Luteinizing hormone surge leads to disintegration of endometrium. This is again wrong. It cannot disintegrate. The high concentration of LH, how can it disintegrate? The LH is responsible for maintaining the corpus luteum, and if the corpus luteum is maintained in that condition, the concentration of progesterone will remain high, and because of that, what will happen? The endometrium will survive and endometrium will remain intact. So this is wrong statement. So statement A and B are true only. So option number 1 is true. All living members of class Cyclostomata are ectoparasites. How? Why? See, when we talk about ectoparasites, oh sorry, it is endoparasite. This is ectoparasite. When we talk, they have circular mouth and they have 2 rows of inwardly directed homodont teeth inside. Okay, they stick to the body of large fishes, like we talk about that whale, or the not whale is not the fish, it is mammal or shark. And they attach to the body wall of the large fishes. Okay, and they suck the nutrients from their body, and that's why they are considered to be the ectoparasites. So fourth option is the correct option. Now let's move on the next question. Now question number 142, assertion: it is all vertebrates are chordates, but all chordates are not vertebrates. It is the correct statement. It is written that all vertebrates are chordates. Why? They have had that notochord and nerve cord in the early stage, that is replaced by the cartilaginous or bony, that notochord and that vertebral column, and the spinal cord is that nerve cord is replaced by that spinal cord. That is why all the vertebrates are what? Chordates. Okay, so here in the reason, the membrane of phylum, subphylum vertebrate possess notochord during the embryonic period, because they have this structure in the embryonic stage. That is why they are chordates. So the reason is again the correct explanation of assertion. So both the assertion and reason are true, and R is the correct reason, is the correct explanation of assertion. This is the correct option. First, identify the statement that is not correct. Okay, so this is the question from that structure of antibody. Okay, so the antibody looks like that. Okay, so here, so this is like, this is N terminal, this is N terminal, this is C, this is N, this is N, this is C, C, C. Okay, this is the heavy chain, this is the light chain. Okay, now let's move on the question. Identify the statement that is not correct. Each antibody has 2 light and 2 heavy chain. Is it correct? Yes, it is. The heavy and light chain are held together by disulfide bond. Obviously, it is also correct option. It is given in the diagram here. I have drawn here. Antigen binding site is located at C terminal. No, it is located at N terminal. The variable part of that heavy and light chain is present towards the N terminal. So this one is the wrong statement. Constant region of heavy and light chain are located at C terminal of antibody. Okay, so it is the this part FAB is this one. This part is called FAB. fragment, constant fragment. These are the constant fragments. Got it? Okay. So the variable chains are present towards the end terminal of the polypeptide chain. Okay. So third will be the correct option for that question 143. Now, silencing of specific mRNA is possible via RNA interference because of its complementary nature with the target mRNA. Because of its complementary, what happens that microRNA or siRNA forms it pairs with that target mRNA, and after that the target RNA is disintegrated. Okay, so because of that complementarity, that complementary double stranded RNA. Okay, so first option will be correct. That mRNA, it targets the mRNA, not the transfer RNA. So complementarity double stranded RNA. Okay, because of that it disintegrates, it stops the synthesis of protein. Okay, in the process of translation. Now, okay, the first menstruation is called, a very easy question, it is called the menarche. Okay, starting of that menstrual cycle is called menarche, and the last menstrual cycle is called the menopause. Okay, so now let's move on the next question. Okay, there is again the two lists of 1 and 2. Heart, kidney, what is it given? Okay, so in the list 2 they are given the hormones, whatever is secreted from this organ. Heart, atrial natriuretic factor is secreted from the wall of auricle. So A will be connected with third, and here we have only 4 option, here 4. It is very easy. Okay, kidney secretes what? Erythropoietin. So B second. Gastrointestinal tract secretes what? Secretine. Okay, so C will be connected with 4, and adrenal cortex secretes what? Aldosterone is responsible for reabsorption of yes, sodium ion chloride ion from the distal convoluted tubule, and that's why again the water is also reabsorbed. Okay, so here the option 4 will be correct. Now let's move on the next question. The protein part of enzyme. See, when we talk about holo enzyme, what we see, holo enzyme is formed by apoenzyme. It is formed by apoenzyme and cofactors. Okay, we have three different type of cofactors. That first one is called that prosthetic group, the second one is coenzyme, and third one is metal ions. Okay, so here when we talk about that apoenzyme, this part is proteinaceous part, this part is proteinaceous protein part, this part is apoenzyme is protein part. So the protein part portion of the enzyme is what? The apoenzyme. Third option is correct. It is also a very easy question, you can solve it very easily. Now let's move on the next question. Okay, sweet potato and potato. See, the sweet potato is the modification of root, and potato is the modification of a stem, but both works similarly. The function is same because both stores the food. So what it shows the analogy. The origin is different but the function is same, and this condition is found in convergent evolution. They have originated from different sources but they are performing similarities, they have similarities in function. That's why it is convergent evolution. So the third option, analogy and convergent evolution. Analogy and convergent evolution will be the correct option for it. Okay, now let's move on the next question. Okay, after maturation in primary lymphoid organ, the lymphocytes migrate from interaction with antigen to secondary lymphoid organs tissue. Like, what are the secondary lymphoid organ? They just asked that. Okay, so thymus and bone are what? The primary lymphoid organ. Okay, the thymus and bone are the primary lymphoid organ in the human body. Okay, and spleen, lymph nodes, and Peyer's patches are what? The site of interaction with the antigens. They are considered to be the secondary lymphoid organ. Okay, so the answer will be C, D, and E. C, D, and E. The fourth option here will be the correct option. Now let's move on the next question. Okay, consider the following statement regarding function of adrenal medullary hormone. Adrenal medullary hormone, that means they are talking about adrenaline. Okay, they are talking about adrenaline. Okay, so what adrenaline do? It causes pupil dilation, not constriction. Okay, in emergency situation the pupil needed to be dilated because of excessive amount of light is needed to be entered inside the eye. That is why we can watch easily, very easily. Wide gaze is available on that time. Okay, so this is wrong. It is hyperglycemic hormone. Yes, it breaks down the stored glycogen into glucose molecule and is released inside the body for quick energy supply. It causes piloerection. Yes, it is responsible for piloerection. It increases strength of heart contraction, as we have discussed in the previous question already is this point. Okay, so the correct answer will be B, C and D. So B, C and D only, that second option is correct. Okay, now let's move on the next question. Why can't insulin be given orally to diabetes patients? See, the insulin is protein. Insulin is protein. If you directly give it by orally, the digestive enzyme will digest it, isn't it? So here you can see human body will elicit a strong immune response. No, because it is very much similar to the protein of the body. How can it will be elicit the immune response? Okay, it will be digested by gastrointestinal tract. Yes, second is the correct option. Okay, now let's move on the third one. Next question. Okay, so which of the following hormone released from the pituitary is actually synthesized in the hypothalamus? Okay, in the class, hello, in the class, students, teachers must have discussed that there are two hormones, that ADS, antidiuretic hormone, and the oxytocin are synthesized in hypothalamus but are secreted from the posterior pituitary. So let's see the option. Luteinizing hormone, wrong. Antidiuretic hormone, yes, correct. Follicle stimulating hormone, wrong. Adrenocorticotropic hormone, wrong. So the second option will be the correct option. Okay, let's move on the next question. Role of the water vascular system in the echinoderm is what? It helps in that respiration and locomotion. Is it correct? Yes, it helps in respiration and locomotion. Excretion and locomotion, no, it is wrong. Okay, capture and transport of the food, yes, it is correct. Digestion and respiration, no, it is wrong. Digestion and excretion, no, it is wrong. Okay, so it is directly the statement taken from the NCERT. So this water vascular system is responsible for respiration, locomotion, capturing the prey and transport of food. Okay, so the A and C only. Is there any option? Second, A and C only. Second is the correct answer. Okay, now let's move on the next question. Which of the following type of immunity is present at the time of birth and is a non-specific type of defense? They are talking about non-specific type of defense. That means they are talking about about innate immunity that is already present at the time of birth. So the second option is directly the correct answer. Now let's move on the next question. In frog, the renal portal system, there are three types of portal system found in the living organism, in the vertebrates when we talk about. Okay, first one is the renal portal system that is found only in the fishes and amphibians. Okay, so in fishes and amphibians, what happens? From the hind part of the body, from the hind leg, from the tail part of the body, the blood is directly transferred to the kidney. Blood is directly transported to the kidney for filtration first, because they have used large part of that hind body part. Okay, that is why. So in renal portal system, what happens? The blood is directly transferred to kidney, where it is the kidney and lower part of the body is the correct option. Fourth option will be the correct option. Now let's move on the next question. Okay, which of the following enzyme are not essential for gene cloning? In gene cloning, we need to cut that DNA. Obviously, we need that restriction endonuclease. DNA ligase, yes, we have to add the DNA, so we'll need the DNA ligase. DNA mutation, no, we do not need any type of mutation in that, so we will not use it. DNA recombinase, no, we will not use that DNA recombinase. It is used at the time of gamete formation when the meiotic division occurs in the first prophase. First liptotin, zygotin, packaging, diplotin, dikinesis. You must have studied all these stages, and then this stage what happens, that the chromosomal part is rearranged, so there it is needed. So we do not need that, we are not performing that process. DNA polymerase, yes, obviously we need that, because we need to amplify the DNA polymerase in the process of polymerase chain reaction. So we need that A, we need that B, and we need that E. Okay, so A, B, and O. Okay, not needed, na? Not needed. So C and D is not needed. Okay, so first option will be correct. Okay, I'm going with the flow. Now, here, frog respire in water. See, this statement is directly taken from the NCERT. Okay, there is some mistake in it, and we have to find it out. The frog respire in water by skin. Yes. By buccal cavity? No, this is not correct. And on land can respire by skin? Yes. By buccal cavity? Yes. And also by lungs? Yes, it is correct. So what we have to find out? We have to find out the mistake. We have find out that by in water they cannot respire by buccal cavity. Okay. The statement is true for water. Yes. Is it true? No, it is not true for water, but false. So it is true for land but wrong for water. Let's find out the option. False for water, yes, and true for land, yes. So third option will be the correct option here. Okay, so let's now move on the next question. Twins are born to a family, okay, that live next to you. Okay, the twins are a boy and a girl. Boy and girl, that means there is genetic difference. Okay, which of the following must be true? Find out that. Because they are two different gender, that means they have formed from two different ovum and zygote. Okay, so they are not they are fraternal, that means they are dizygotic twins. Let's find out the option. They are monozygotic twins. No, if they would have been that monozygotic twin, then gender must be same. Okay, they are fraternal twin. Yes, they are fraternal twin. They are not monozygotic twins. So second option is correct. Fraternal twins means non-identical twins. Okay, now let's move on the next question of zoology. Okay, here again we have the list 1 and list 2. Okay, progesterone. See, here you can get confused between this progesterone and relaxin. Both are actually secreted from ovary, but if we have to specify, then we can specify that relaxin is secreted from corpus luteum and progesterone is secreted from ovary. Got it? Relaxin is secreted from corpus luteum and progesterone is secreted from ovary. Okay, now melanocyte is melanocyte. Okay, melanocyte, that means pars. Melanocyte stimulating hormone is secreted from pars intermedia, and catecholamines is secreted from adrenal medulla. Catecholamines are called the biologically active amines. Okay, the adrenaline is the catecholamine. Okay, now let's connect the option number 1 A with 4. A with 4 we have two options, B and second. A with second. A with second we have one option, and B with 4, okay, and C with 1 and D with 1. So third option will be the correct option. Now let's talk about I think this is the last question from zoology. The blue and white selection markers have been developed with different recombinant colonies from non-recombinant colonies on the basis of their ability to produce color in the presence of chromogenic substances. As we have already discussed in a question, that Z gene produces beta galactosidase, and what does it do? That X-gal is converted into blue color pigment. Okay, now let's study that statement. The blue colored colony have DNA insert. Will it be possible? No. Why? if the DNA has been inserted inside that jet gene, there would have been that insertional inactivation. So statement A1 is incorrect. Okay. The blue colored colony have DNA insert in the plasmid and they have identified as that recombinant. This is wrong statement. The colonies without blue color have DNA insert. Yes, the colonies without blue color have DNA insert because of insertional inactivation in the plasmid and are identified as recombinant. This one is the correct statement. So statement 1 is incorrect and statement 2 is correct, option number 4. This is all about zoology. And see students, this paper is bit more tough. If you compare it with the previous years, 5 years of previous years question, PYQs. Okay, so don't take any tension, just relax. The cutoff is going to get down. That is 101% sure. 101% sure. Okay, so we'll continue with the physics and chemistry paper. So thanks from my side. Thank you all, and just best of luck for your results. Okay, so we are going to start the discussion of chemistry paper. We have Pallavi ma'am for this. Welcome Pallavi ma'am, and let's start the discussion. That chemistry.
Zoology was fine.
Zoology was a bit more tough
Okay.
in compared to that last 5 years.
Even in chemistry also, this is tougher way. Even I am finding this book from past 4-5 years. This time it is like tougher and lengthier. Okay. Okay, so let's start with the chemistry portion. Yes, I'll fix it. Okay, so okay, so the question is in continuing. Okay, so let's start doing the chemistry section. So here the ratio of the wavelength of the light absorbed by hydrogen atom when it is happening from 2 to 3 and there is 4 to 6. So we have direct formula. That formula is that 1 by lambda would be is equal to RH 1 upon N1 square minus 1 upon N2 square into Z square. So when you are going to put this value 2 and 3 in place of N1 and N2, you will get the lambda 1. And in second case, you will get it, that is, instead of N1, you can put 3 and 4. So you will get the two equation, and on solving these two equation, you will get the answer. That is the 1 by 4. It should be the answer. Okay, let me just check with the original question in the copy. Just 1 minute. Yes, so your answer should be the 1 by 4. Let's move to the next question. They are saying which of the following statements are true? Okay, so like gallium, here they are saying gallium that has the very high melting point and cesium having the very low point. So basically that gap is not that great. Gallium is 29 point something degrees Celsius and cesium is around 28. So hardly that 1 degree Celsius difference, but here they are saying it's very high melting point and low melting point. So not considering is the right one.
On Pauling scale, the electronegativity of nitrogen and chlorine are not same. Yes, that's right. It's not same. Where argon is 18 number, potassium is 19 minus 1, 18. This is 17 plus 1, 18. And that is 20 minus 2, 18. And sulfur is, that is again 18, 16 plus 2. Okay, so this is isoelectronic species. C is the right option. And the correct order of the first ionization enthalpy. Okay, this is not right because magnesium is half full. So obviously this will come first, then the aluminium. And the atomic size of the cesium is greater than the lithium and rubidium. Yes, that's the right answer. So we will check with this. So we have a, that is E and C should be the right. C and E should be the right according to the option, and we will mark that. Okay, let's move. So why we are not writing B? Because in none of the option that is contained B, C and D. So we are just matching with the B, C, E. We are just matching with the two option. Okay, here we have, that is the match the column. Okay, so this is the group based question. So let me find out that group based question where it is. Okay. So that is the 3, 4, 1 and 2. This is matching salt analysis question 3, 4, 1 and 2. That should be the right answer for this. Yeah, sir. So we have Gaurav sir also there. So he is also going to talk. I think 2 mic is not there. हाँ, we will share. ऐसे कर लेंगे sir. So these questions, sir, we have to careful because here the different, I think that group or the booklet number is different, so we have to check with the right answers. Okay. Yeah. Let's move to the next question. Yeah, sir. So can you help me out for this to get the right answer?
Yes, okay. So students, in this question, if you see, the question is like a bit conceptual, the organic chemistry question. So we are having the addition reaction of HBr on alkene, and we are using the peroxide here, benzoyl peroxide, if you see. So obviously it follows the anti-Marconi powerful, so addition of Br will come here, and then H will come on this carbon. So if you look at the first step, we get here a 5-member ring. Br will come here. Cn we have, CH3 we have here already, and H will come here. After this, we have reaction with KCN, so there will be substitution of the nucleophile Cn minus here, and it will lead to the formation of cyanide, which will come here, and CH3 here. And then we have the reduction with amalgamated sodium with ethanol. So it will convert this Cn into amines. The product will be CH2, NH2 here, and CH3 as we have already. So this is the possible product. The product would be option 1.
Yes.
This was quite conceptual, right?
Yes, very conceptual. Here the anti-Marconi off and that SN2 mechanism and reduction, everything is included. Quite good question. Okay, let's move to the next question. Okay, so energy and radii of the first Bohr radii they are asking. So we just need to apply the direct formula. So energy would be is equal to minus 13.6. Okay, they were writing in the joule, so let's use in the joule. That should be is equal to 2.18 10 to power minus 18 Z square by N square. That is in joules. So for helium and lithium, you can put the value and get the answer. And radii would be is equal to, that is 52.9 N square by Z. Okay, so N square by Z, when we are calculating for helium and lithium for both, so we are matching with the answer that is okay, let me check with the answer because this is different. Yes.
So you just have to substitute here the value of Z for different ions accordingly. The answer in this case would be option 3. For lithium, Li2+ will get minus 90.
Yes.
0.62, so option 3.
Oh, okay, sir. I think here the slightly difference because they jumbled the options. So, yes, yes. So 19.62 10 to power minus 18, that is the right is matching. And then 17.6 picometer. And then helium is matching with the 8.72 and 26.4. So actually option one. So everyone need to, those, no matter which group paper you received, you have to check with the values. Yes.
The only difference here is minus 18 and minus 16.
Yes, yes, yes.
That's all.
Yeah.
Everybody we can make.
And this is actually jumbling their options. Yes. So next one we have, which of the following are paramagnetic in nature? So for paramagnetic, that we have to check it. That is the weak field ligand or strong field ligand. So here, if we are checking with the nickel is number 28. So that is become 4s2 and 3d8. But the oxidation number is +2, so you can remove these 2 electrons from here. It's a weak ligand, so obviously they are not forcefully pair up and it's become paramagnetic, right, sir? And here the carbon monoxide we have, which is a strong field ligand, and nickel is having the 0 oxidation number. So this get forcefully paired up, like it's become 3d10 and 4s0, so diamagnetic. And the same thing is gonna be happen here because cyanide is also strong. Water is weak here. So between A and D could be the right answer because this is triphenylphosphine is also the strong field. Yes. So that's why your answer should be A and D. Okay, so option 3 would be the right answer. Yes, sir. Please discuss the next question, sir.
Okay, so like you discussed, this question was like quite easy, simple concepts of coordination chemistry we are discussing here. Now the next one is factual inorganic chemistry they have asked. And the question, the statement 1 we have, like nitrogen that can form ammonia, arsenic can form arsine, right? Arsenic. So arsine is basically the hydrides of the arsenic, the element that you have. So obviously that's true because we have NH3. Similarly, arsenic also forms the hydrides, which is the formula we have AsH3. So this is basically factual, right? You have to know this. So statement 1, I think it is correct. Statement 2, you see antimony cannot form antimony peroxide. So antimony peroxide, it won't form obviously. So statement 2 is incorrect, I guess, right ma'am? Yes. Statement 2, right? Right. So that's the question is here. It is factual questions. It's like the the information you should have, and the answer would be here. Statement 1 is correct and 2 is incorrect. So I think I'll go with option 3. Is it, ma'am?
First is correct and statement 2 is
Incorrect. Okay. We'll move to the next question now.
Yes.
Okay. Right. So which of the following electronic configuration belong to the main group element? Main group element, there is no exceptional configuration we are going to have. So you have to see for a given elements, the configuration which is given here, it is actually the same as we have for the elements belong to the same group. There is no exceptional configuration we are going to have. So obviously the A you see neon 3s1. This is basically sodium, right? So this one is correct. We don't have any exceptional configuration here. Similarly, if you see here, which one? E, right? If you see this E here, this is also 6d2 7s2, 7th group element it is. So I think option A and option E will have the right configuration here, the electronic configuration. We have the main group elements. Option D, you might have some doubt while solving this. I think C also we have, right, ma'am? I think option C we have. I took the wrong one.
This field will not show because this is the F block elements.
Yes, ma'am.
Yeah. So this is the F block element, so we can eliminate. And the main group element, I think we consider S block element only. So we would go with this sodium is 11 number. It's visible here.
C.
Yes. Yeah. Option C is yes. Option C also.
This we can't.
This because total is 18 plus 10 plus 1 is coming copper.
29.
Yes, and that is 29. So D block elements, and these are the valence electron
And
for
what
this.
about option B, ma'am?
Sir, here also that is the valence electron is coming in D. So that's why it is also the belongs to the D block compound. So this is D block, this is S block, this is P block, and that is C, it's F block. So we would go with the A and C.
A and C. So correct option in this would be A
That
and
is,
C,
yeah,
option
option
2.
2. Yes, yes.
Yes. Next.
Yes. So Dalton's atomic theory could not explain which of the following. So we know that it's basically conservation, it depends upon the conservation of mass. So conservation of mass is perfectly fine. Constant proportion is perfectly fine. Multiple proportion is also perfectly fine. The gaseous volume, I think, sir, this is given by the, that is Galuka. Yeah, Galusa.
So I think the correct answer is option 4.
Yeah, right, right. Yes. So consider the following compound, the oxidation state of the underlying. Okay. So this is superoxide, but potassium always having the +1 no matter what. Yes. And this is peroxide linkage, so -1. And this is, it's a 6 if you are finding. So that is H2SO4. This is +1, X and -2. So when you are doing the addition of this, you will get the 6 number. So we will have the answer +1, -1 and +6. Answer should be
So I
A.
think this was an easier one, right?
Yes, yes.
Straightforward.
That's so many. Few questions are only the easier, but maximum question are taking too much time, right, sir?
This question.
Okay. So if the half-life for the first order reaction is 1 minute, then the time required for 99.9. So sir, वैसे तो we can use it the short trick, but but it takes too much time because we have to reach the 99 point. So let's use the formula. K would be is equal to 2.303 by T. We have to find out log of initial concentration, which we could assume 100, and that should be 80, which we could assume 100 minus 99.9. And K value, we can put it, that is 0.69 by T half. And T half is already given, that one. So when you are keeping this, the T, you will get the 10 minutes. And there is no need to change this minute into the second for calculation because our answer is coming in only minutes.
So we have one also approach in this. We can directly also use the time required equals to, if it is 99.9% of
Yes.
completion, if you remember, 10 times 50.
Yes, yes, that is the, yes, yes, that is the direct one, yes. Please take this one.
Now the next question is, we have to find out the correct order of wavelength of light absorbed by the following complexes. Okay. So wavelength of light for the following of, we know energy is directly inversely proportional to the wavelength we have, right? And we have different ligands here. So we have to go with the strength of the ligand. So clearly the strength of the ligand, we know Cn minus is a strong ligand we have. The strength is more than NH3 and which is more than water, H2. So clearly if the strength is more, that means the energy delta O for this one, it is octahedral complex, will be maximum here. And hence the wavelength in this case will be minimum. So wavelength of Cn minus will be minimum, NH3 will come in between, H2O will be maximum in this case. So this is minimum. So one thing for sure, B is minimum here, right? And then we have A, and then C and D. C and D, how can we explain it? This is tetrahedral, this is octahedral.
Yes, in general, what is happening that titanium. So here the answer should be B, C. The copper having the lesser energy to more wavelength it should come.
Right.
Yes. So
B is minimum, A is there, and then we have C maximum, right? C maximum,
and that should be A and B.
So here also you see, if you look at the options here, options are also not very straightforward.
Yes.
This two, though, we are very much sure it is going to be right, but again C, D you have to sure which one is maximum, which one is.
Can you see that there is a the catch? They have given only one option, B and A. In the
Right.
So if you are aligned with
Yes.
this and this,
We can eliminate.
so if suppose you are not aware about these two, you can.
Still we can. That's fair. Yeah. Continue.
So the
Okay.
next one, which one of the following compound can exist as cis and trans? So we know that the condition should be the different substrate and the restriction and rotation. So in that case, so we don't have any different substrate. So first one is gone out, but X2 okay. So here, okay, it's not cyclic, sorry, it's an open chain. So 1, 2, 3, 4, 5, 6, and that is the 2-methyl hex 2 methyl. So here, this is also not correct because here the substrate has become same. We will have the cyclopropane now, cyclopropane 1, 1 dimethyl. Okay, there is again no option is there. So we will left only with the one option, hexane. Okay, hexane 1, 2 dimethyl. So what do you think, sir? That can we make it like this?
Yes, ma'am. And keep it same side, and then one will be above the plane, other one will be below the plane.
Yes, yes. So like this is the, if it is a solid line, this should be the dash, or both should be solid and both should be the dash.
That is the possibility.
So we will have the answer.
Option 4. Next. Okay, so this one, you see the phosphoric acid. You all know the formula for phosphoric acid is H3PO4. If you do not know the formula, then also can we do, ma'am, this?
Yes, sir.
Because three constants are given.
Yeah,
so
right.
tribasic acid we have to consider, right?
We need to make sure that all 3 hydrogens should be polar.
Right.
Otherwise, it could be
Exactly, exactly. So here we all know that H3PO3, PO4 it associates as H plus and H2PO4 minus. Right, so this association is Ka1 we are assuming. Clearly we have a negative sign here. So if you want to take one more H plus out from this, that is going to be relatively difficult. So this will be again H plus HPO4 2 minus, which further dissociates and gives the third H plus ion, HPO4 2 minus, which converts into H plus and PO4 3 minus. This is given as Ka1, Ka2 and Ka3. So clearly here, this is maximum Ka1 because the dissociation in first case will be easier. Then we have Ka2 and then we have Ka2. So this C is to definitely right. We have options here. Could you explain on how we are getting the other cases possible?
Yes, sir. So let's take it. This is, we know that we learned in the chemical equilibrium, if you need to identify the overall one. So you need to add up all this such thing. And when we are adding, the overall should be is equal to the product of this. And so when we are taking the log thing, so log K would be is equal to log Ka1 plus log Ka2 and plus log Ka3. So it's very obvious that we are matching with the first option, right? A, yes.
Any other statement in right now?
Yes, sir. H3PO4 is stronger than this one. So I think everybody learned in the chemical equilibrium, that is the common ion effect. So if you are seeing, so this is creating the common ion effect for the first one. So sorry, so reaction is going into the backward direction. So obviously that ion is not that get dissociated like a first one. Again, one more time, the common ion, this also will go backward.
And that's also the reason we have this order.
Right. So that's how we can say that H3PO4 is a stronger, because for first case there is no common ion, but for the second and third case we had a common ion. So based on that, your A answer is right, B is right, and C is right. This is not right. So let's A, B, and C. That is the
Option
fourth.
is the right.
Right, four is the right
This was,
answer.
I think they usually do not ask.
Yes, in the
polybasic acids in NEET.
right.
This time they have asked. So yeah, paper was good.
This paper was good and
Next question.
yes.
Okay, so we have which of the following reactions does not give benzene as the product? So this was obviously the direct question we have. You know, this is soda lime we are heating. This is soda lime decarboxylation. Obviously, it will convert into benzene. This is soda lime decarboxylation. This is simply the aromatization reaction we have here. Molybdenum trioxide we are using. It also converts this into benzene. This is red hot carbon. Yes. So this also converts this into benzene. This one gives you phenol. It is a reaction of warm H2O with benzene diazonium chloride. This converts into phenol. One of the preparation method of phenol also here.
That's very important for the other competitive examination also. So I'm wondering, this time NEET has really prepared the good question paper, and a student must face the time problem, like time management is going to be probably
I know
little
many
difficult.
students have this complaint also because they have clearly mentioned that benzene diazonium salt is not in the syllabus.
Okay.
Diazonium chloride they have removed actually, but yes, we have done in the class by the way, some reactions we have.
So that's good for the PW student. Yeah. Okay. So we have this question now. If the molar conductivity of weak monobasic, that is the weak acid, is 90, so the extent of the dissociation will be. Okay, so degree of dissociation, we learned it in the this thing, electrochemistry. That is the molar conductivity at C concentration divided by the molar conductivity at infinite con. So at C concentration is already given, that is the 90. And this we will apply based on the Kohrash law. So we know that HA is going to produce one cation and one anion only. So the molar conductivity at infinite dilution, the conductivity produced by the cation and conductivity produced by the anion. So they have given the value here. Cation is 349. I'm keeping directly. And anion is 50.4. And on calculation, you will get it. That is 0.225. You will get it. Yes.
So, ma'am, why they have given this concentration there, 0.05 mole per liter?
Yeah.
Just to kill the time over.
Basically, degree of dissociation is the number of particles. So whether, no matter what concentration they are giving, it's all depend upon how much ions has been produced by that. So it could be the weak electrolyte or strong. And basically, degree of dissociation, we are comparing with the some extent of the dissociation divided by the 100% dissociation. Yes.
That is not required. Actually.
Yes, not required. It's just for the confusion.
Just for the confusion, yeah. Okay, so the statement is the hypothetical, this was, I think, the simple one, right, ma'am? Hypothetical diatomic molecule with bond order 0. Obviously, bond order 0, that means the molecule does not exist.
Quite common sense based question they have asked.
Exactly.
Diatomic and the bond order is 0.
Right.
Yes.
So it is not possible. So statement is obviously right. Hypothetical diatomic molecule with bond order 0, quite stable. The statement is correct. Not stable, no, incorrect. I'm sorry. Okay, incorrect. As bond order increases, bond length decreases. That is correct only. We all know carbon-carbon single bond is, greater than carbon-carbon double bond. Carbon-carbon triple bond. If you have the bond order 3, 2, 1. So second statement, as bond order increases, bond length increases. This is also incorrect because bond length has to decrease. Here, hypothetical diatomic molecule with bond order 0, that is not possible. It won't exist, so it won't be stable. So I think both statements are incorrect. Right?
So we have to check carefully here. Both statement 1 and 2 are
False.
Option B. 2 could be the right one.
Correct. Yeah.
Okay, this is something garbad question. Out of the following complex compound, which of the compound will be having the minimum conductance in the solution? So I think every single student learned the more number of ions, more conduction. Here we can see that is 1 and 3, that is the ions are 4. Yes. So here we can see this is 1 and this is 1, so number of ions is 2. Now here is a single thing, this is also single thing. So they both are non-electrolyte basically. What I'm finding, sir, they missed out some chlorine concentration here because the 3 chlorine they have to use it, but here they forgot to use that chlorine outside. So I believe for this question, 1 and 2 both can be the right answer. Yes.
I think they'll give you grace marks in this or what?
Probably they have to consider that.
But as of the question is given, both answer could be right. One
yes, yes. 1 and 2.
What we are assuming that there should be a chlorine here, which is missing out in the question. So I think when the NEET key will come, they will give answer accordingly, or they'll make some changes.
Yes. Or those students who put 1 or 2 maybe
they should get the marks. Okay. Yeah. Next question. Okay. This was the question from chemical bonding XeO3 XeF2. We can find out hybridization easily. We know the method how to find out hybridization. I am going a bit a bit direct in here because I know you all are looking for the answer keys now. You're not into the solution.
They must be very tired also.
Exactly.
From the morning they were busy in the NEET exam.
Exactly. So FeF6, this one is pentagonal bipyramidal. This is sp3d3 hybridization. XeF2 is sp3d. We know it is linear. The shape is, and the steric number is 5 here. This two information, if you have clearly, sp3d is going to be the first one, linear it is. sp3d3 is going to be the third one, which is distorted octahedral pentagonal bipyramidal. XeOF4 is which one, ma'am? It is fourth one, is it?
Yeah,
or shall I
that
check?
should be- C should connect with the
Fourth one, she is the fourth one, and first one is the second.
Right.
Okay, I'm assuming you all know how to find out hybridization and all, so we're not going into detail of this solution. The correct answer is going to be A is 2, B is 1, A is 2, B is 1, C is 4, and D is
Right.
option 1 is the correct answer. Okay, next.
So next question is a carbon and hydrogen combining and forming the methane, and del H is minus 74.8, means this is the exothermic reaction. And we all learned it in the chemical kinetics. If the energy diagram we are drawing for the exothermic, that should be like this. So let's do the 50-50, just like KBC. Here, this is exothermic. So this seems exo, this is endo, this is exo, and this is endo. And we know that the del H value is nothing but the HR and HP gap only. This gap is called the enthalpy of reaction. So let's find out. Here they have given activation energy, so this is wrong answer. Your answer should be this one.
This value is the activation energy, right? This value
Yes, this
is the
value.
activation.
Yes, activation. So here also we can find this is the activation energy. And they are asking only about the delta H value. So we will mark number 1.
Okay, this is also correct. Yeah. So basically, humidity is what? Humidity is nothing but solid in. Okay, I'll do like this. Alloy, we all know. Alloy is the mixture of solid and solid. So B is going to be 1. Amalgam, we know it is the alloy of mercury. So we must have liquid in solids. So liquid in solid, C is fourth option. Smoke is solid in gas. Smoke is solid in gas, and then humidity is liquid in gas.
Now these days Bangalore is having too much
Humidity, exactly.
Because
This though you should know about.
yes, so that's why liquid in gas.
Okay, so the correct answer is
We can match the this is the
A is
second
2,
one.
B is 1,
Second one. Only
C
one
is
is
4. I think this one, yeah. This
So for such type of question, it's good if we are matching with any two. The
Any two we
option are
can
matching.
yes. Okay, ma'am. The correct order of decreasing variance.
Basic strength of the given amine. Okay, we have to draw the formula. However, we know that amine's basic strength is obviously depends upon the aqueous state and the gaseous state is different. So let's find out N-methyl aniline. Okay, so we have N-methyl. This is the aniline, and I'm connecting the methyl group. And methyl group is connected with this, and benzamine is nothing but the aniline.
Benzene.
Benzenamine is nothing but the aniline, and ethanamine is a C2H5NH2. And then we have, that is the N-ethylethanamine. Okay, second degree, that is the C2H5 and C2H5. So I believe, sir, if the bulky behavior is increases in the aqueous medium, so the basicity is getting down.
Yes.
Yes. So in such type of cases, that should be the highest basic.
Yes.
And then it should be aniline, so we know that then lone pair is participating in the resonance.
Yeah.
Here also it's a plus R effect, that is the plus I effect
Plus
is
I effect.
effect is there, but still the basicity is gonna be decreased because lone pair is gonna be involved in
In
the,
comparison to this two.
yes, yes.
In this two, we can say this one is more basic.
Correct. So how we can write the order? That is one should be on the top, then this is number 3, and that should be number 4.
So the option that we are going to
Yes. So N-ethylethanamine, then ethanamine, then N-methyl aniline. Okay. So whomsoever that PPT we are using, actually, the student maybe mark it the right one,
Right,
because
yeah.
there the sign is there. Yes. Okay.
Okay, next is simple questions from the mole concept. We have to find out the number of atoms, and we all know to find out the number of atoms, you have to find out number of moles, molecules if it is there, into Na, into number of atoms in one molecule. So I'll just do the quick calculation for option 1, like the first one that we have, which is 12 gram of Na2CO. So if you find out the number of moles here, this is going to be the given mass divided by the gram molar mass 106, which is here 2. This is number of moles, so number of molecules would be 2 into Na. If you don't do also, it's fine, because this is going to be same in all the 4 cases. This is the number of molecules, and one molecule, you see, it has how many atoms? 2 sodium atom, 1 carbon, and 3 oxygen. So we're going to have 6 atoms here. So this is the number of atoms in the first one, which is Na2CO2. So basically, you have to find out moles into number of atoms in one molecule, right? So this is going to have 12, right? So I assume you can do this calculation now. The correct option is going to be, I think, option. This is correct. This is correct, right, ma'am? 12, and this was not this one. I think NaOH.
That is Na2O also having the
12 better.
12.
Because the options are jumbled here.
Yes.
Okay, I have done the calculation for you. You can just cross check this
Yes,
accordingly.
yes. We have, along with the student, we were also solving the question paper.
Yeah.
So that's why we were checking this. So here also the 12 is coming. Here
Okay,
also the
12
is
atoms,
coming.
12 atoms.
And this is the calculation we did, sir. And here is the 18.
18.
Yes.
We're getting 18.
12 by 2 into 3. With hydrogen is
Having 12 atoms.
yes, 12. Yes.
12
We
by
are
2
getting
into
the 12.
So I think these three are right.
Okay.
How much in this we are getting now? 20 to 40 grams. 240 divided by 40.
240 into 3, 18. 18
So we are going to get 18. So obviously this one is not. It was going to have 18 atoms. 12, 12, 12, and the last one
Last case we are getting 15. 220 divided by 44
44 into
3.
so 15 here.
Here the 15.
So answer is A, B, and C. D, sorry, A, B, and B. A, B, and B. Option. So this was the wrong one.
Okay, that's wrong. Either the 1 and 3, both of them were wrong.
Right. Okay.
Okay, next question.
This was the like.
Yes.
You know or you do not know. You can't do anything into it.
Yes.
Right.
So I have taken help from the bio sir
Okay.
and bio ma'am as well. So B12, according to the B12 deficiency, the anemia will happen.
Okay. So A is 4, right, ma'am?
Yes.
A is 4.
And the vitamin D is rickets.
Rickets. So B is going to be 3.
So 4 and 3, where we are matching 4 and 3, we are matching here and here.
Okay. Again, we have 2 options.
Yes. And vitamin B2 is okay. I'm good in bio, so I have a slightly
Kilosis.
here. That is
Kilosis.
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 Yes.
So B2 is this. Okay. So don't mind the pronunciation. Okay.
Yes.
So this is 2.
So let's
So we are going to have
4, 3, 1, 2.
yeah,
4,
4,
3,
3,
1.
1. So option B, right?
Option 2.
Okay. Okay.
So the correct order of decreasing acidity of the following aliphatic acid. So we know that more minus I effect. So more minus I effect, or maybe the more withdrawing group can create the more acidic behavior. So these are the carboxylic acid family. So it's very direct answer. Formic acid is on top, then acetic acid, and this is pretty bulky here, and that is the tri-substituted carboxylic acid, huge plus I. So we can directly go with the third
So I have
number.
this was the
Easier one, yes, 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 yes.
Next.
Okay.
Again, this one is the factual one. Ferromagnetism, it is the extreme behavior of paramagnetic substance, as we all know, right? So this is the first statement is correct. This is also factual. You should know this. Ferromagnetism, the behavior that we get in paramagnetic substance only. Statement 2, the number of unpaired electrons in Cr plus 2 is same as this one, Nd3 plus. Okay, this one is correct. Cr2 plus chromium plus 2 has how many unpaired electrons? 3 unpaired electrons we have here, right, ma'am?
So chromium plus 2, I think we have to check one more time. Chromium is 24 now.
4s1, 3d5.
4 is 1, 3
Okay. Plus 2 we have. Is it?
Yeah.
Acha, the 4 it is. 3d4, 4 unpaired.
We have
My
4.
bad. It's 4 unpaired. And this one, ma'am, Nd3 plus
Nd3, that is the
60 atomic number it is.
yes, around 4s4 and 6s2. So when we are going with the plus P, we can remove the electron from
Plus
the
three.
outermost shell and it becomes 4s3.
Oh.
3 electron. We can remove the ultimate shell electron and 1 from
Correct,
here.
correct. So this is incorrect statement,
Yeah.
right?
So
Okay.
we have 4 and 3 unpaired electrons.
So statement 1 is correct, 2 is incorrect, right, ma'am?
Yes.
So correct answer is 1 true and 2 false. Option C. Yeah. Next question.
So here we have a match the column. Okay, so those who done the salt and
Solution.
solution also can help, or maybe the purification also can. Yes. So we will go that crude oil in the petroleum industry is a fractional distillation, and the glycerol from the spent layer, that is the distillation under reduced pressure. So these two are the aniline in water is a steam distillation, very common example this is, and this should be automatically steam distillation. So B is matching with the 3, B is matching with the 3, okay. And then C is matching with the 1, C is matching with the 1, okay, we got our answer. So A is connected with the 4, B is connected with the 3, C is connected with the 1, and D is connected with the second. That's the right one, yes.
Okay, so this is again a direct a question, basic concepts of chemical kinetics we have to use. The question is given A gives 2B, and the good thing is they have given the ratio of Kf by Kb. So Kf by Kb, the rate constant for forward by backward. It says here the backward reaction rate is higher than forward rate by a factor of this. Can I write this as 1 by 2,500?
Yes.
So this is given in the question, and we know this is equals to Kc. And we have to find out here Kp. So we all know Kp equals to Kc RT to the power delta n. Once you substitute all the value, you'll get the answer. Delta Ng value is 2 minus 1, that is 1 only here. So Kc is 1 by 2500. R value is 0.0831 into temperature is 1000. So this gives you the answer, which is 0.03, right now? Option 3 we get here. Yes.
Answer is 0. So given below the two statement, benzene disonium salt is prepared by the reaction of aniline with nitrous acid at 273 to 280. It decomposes easily in dry state. I think it's this is the very fact-based statement. It's perfectly fine that benzene disonium salt, we need a lower temperature for this. Insertion of iodine into the benzene ring is difficult, and hence iodobenzene is prepared through the reaction of benzene disonium salt with Ki. Yes, perfectly fine. So
Preparation method of iodobenzene.
right, and both statement are right. So we can go with the both correct. So option 1 is correct. Yes.
Okay, how many products including stereoisomers are expected for monochlorination? So I'll write down the product here directly. You'll get here isopentane we have, is it? Okay.
Yes.
So we have this. So one will be if you attach chlorine here, another one if you attach chlorine here, another one if you attach chlorine here, and one more we can draw when we attach chlorine here. So in this case, if you see the stereoisomers here, there is no chirality here, here also not possible. Here we have this carbon is the chiral carbon here, and here this carbon is the chiral carbon here. No, sorry. So chiral carbon we have here, 1, 2, 3.
I would say which we can consider
This
as a
one.
chiral. And this is not chiral.
This is not there.
So we can remove it off by doing this.
Yeah.
It's not chiral.
So for this, ma'am, we are going to have two products.
Yes.
For this one, we are going to have two products, 1 and 1. Correct?
So total is become
6.
yes. Our answer should be
6. Okay, next. Yes, ma'am.
So they have given the correct order of bond dissociation energy of CH mark with this. Okay, if you are checking this hydrogen is connected with the benzene which is connected with the sp2. So not, it's seriously difficult to remove the hydrogen because double bond character. Here, since it's a withdrawing group, so okay, it can pull the electron and release this acidic hydrogen. So this is H plus is out and acetylene ion it will form, which is quite stable because WG group is there. Here if I'm removing this hydrogen, so these two electron will shift and it will convert into the anti-aromatic compound, which is highly unstable.
we are considering deep protonation rate.
Yes, and the second, whatever the reaction intermediate we are forming, that should be stable, either by resonance or I effect or hyperconjugation. So here we are finding when we are proceeding, this intermediate is not at all stable.
Highly unstable.
So that's why CH bond breaking is not preferred by this molecule. So third to certainly is coming behind. So third is here and third is here. In these two, okay, this is the acetylene, quite acidic, we know that. And this carbon can hold the minus sign because of the withdrawing group. So that should be on the top. So third is behind, second is on top, and the first middle. So first option would be the right one.
This was also quite conceptual.
Yes,
You
yes.
don't have to
Yeah,
memorize this.
correct.
Okay, so which of the following compounds does not decolorize bromine water? Bromine water, we know it's a test of unsaturation. It's a direct question. We do not have any pi bond here, so clearly the answer is going to be this.
So I'm finding the examiner has jumbled the answers.
Right.
So whatever the section we got it, here we are finding all answers are here and there only.
Yes, this one.
Okay, so this question, the major product of the following reaction. So I think, sir, we were getting stuck whether we will mark this third answer and fourth or no, we actually second, sir, will flip
Sorry,
that they
second
flipped
or
it.
third one?
Yes, yes. But still we conclude that should be the third one, because when the methyl group is attacking here and on hydrolysis, it will give you the alcohol.
Alcohol, right.
So we will go like this way. Here the CH3 is there, and that should be OH after hydrolysis. And that should be C triple bond N. Even we know that the cyanide is also doing reaction with the CH3 MgBr. So CH3 minus is gonna be attack here. So these pi electron will shift here. So we will get the phenyl here, COH, CH3, this, and this CH3 connected, double bond, and NMGX.
Right.
Later we can do the hydrolysis.
Attenuation.
Yes. So the phenyl CH3, OH, and C, that should be OH, and that should be CH3, and that should be the NH. Now one has to go away, right? So this hydrogen is attacking here and will go as ammonia out. So I think I can make the direct answer here. It will make it C double bond O CH3.
Which is there one option.
Yes, which is there in the option 2.
Right.
But since they have written in excess, so we can attack one more time and convert into the alcohol. So CH3 is attacking one more time and this will convert into the alcohol.
So basically this reaction is ketone and RNA.
Yes, yes. So based on that, we should mark the number 3. So, and let's wait for the final key, what they have given the answer.
In some of the, some places they were giving option 2 also as the correct one, right? But we are assuming this. Let's see what happens in this. Okay, so which of the following aqueous solution we have highest boiling point? Quite simple. This one is, we know the elevation in boiling point delta TB is equals to the Van der factor I times KB into M, right? So here molality and this I value we have to compare. So clearly you see for urea, the value of I into M, if I write here, it is 0.01 into 1. For this one would be 0.01 into, everyone here it is the same only concentration anyways, into 2. This is 0.01 into 3. This is 0.015 into 1. So the one which gives the maximum value will have the highest boiling point here. So this is going to be 0.01, 0.02, this is 0.03. So for our answer will be option 3. The correct. Okay, next one.
Yes. So basically they remove the P block, but I'm finding due to the salt analysis, they put this question. Haber process, we know that it's an iron catalyst. And funny part, sir, have you seen that everything is matching?
Yes. In this
See,
particular
Haber process means iron catalyst, and the Baker process, cadmium chloride with the copper, which they missed, but obviously, yes. And Wilkinson catalyst is this one only, and Ziegler-Nader catalyst is this one only. So they have given everything is matched. Our answer is 1, 2, 3, 4. So 1, 2, 3, 4 options should be C.
Okay, 5 moles of liquid X, 10 moles of liquid Y. Again, a direct question. We know the formula. According to Rawls law, PS equals to, we have P naught A XA plus P naught B XB. P naught B XB. So this is P naught A is 70, 63. 63 times 5 by 15 plus 78 times 10 by 15. If you solve this, the value comes out to be around 71 point something approximately, I'm telling you. But this PS, it is given as 70. That means the pressure which is given here, this is lesser than the pressure according to Rawls law, which means it shows negative deviation. Right, ma'am? Negative deviation. Negative deviation, option 2. This was the easier one.
Yeah, this is the easier one. Sugar X is found in honey. It's a keto sugar. Keto is very obvious. It should be the fructose. Yes. Exists in alpha beta, okay, anomeric form, and it's a laboratory. Quite these two forms are pretty connected with the fructose. So glucose, maltose, and sucrose is out. We have only answered fructose.
Okay, so this is the reduction of ester to aldehyde. We know this LiAlH4 strong reducing agent, it eventually converts this into alcohol, which we do not want. So the ideal reagent that we are going to use here is this one, isobutyl, DBAL. If you remember, we also write it as DIBAL-H, diisobutyl aluminum hydride. So this is the one which, and this we use this to reduce the reducing tendency of LiAlH4 in order to convert ester into aldehyde. We have done all these things in the class. So this is the one that is the answer we are going to have. So option 2 is going to be the right answer. NABH4, ma'am, can we discuss on NABH4 also? It will also convert that into
Yes, NABH4 and DBAL both are the selective, sir. But DBAL is like reducing the ester into the aldehyde. But NABH4 is just reducing aldehydes, ketone, and COCl only. So yes, so we could say that thermodynamically it is not supporting the ester reduction. Yes, lithium aluminium hydride is too active and thermodynamically supporting too high, so convert into the alcohol. Even this BSO4 is there, otherwise it could also be the one of the answer. So that's why we mark it DBAL. So that question is given, the assertion iodine undergo SN2 reaction faster than chlorine. Very right statement. That is the 1 degree. This is primary. Iodine is a better leaving group. So it's a right one. And iodine is okay. They have given the same reason, better leaving group because of its larger size. Perfectly fine. And R is also the right explanation. So A and R are true and R is a correct explanation. That should be this answer, number 1. Yes.
Okay, so we have standard heat of formation of Ba2 plus, and this. So in this case, basically you have to write down the reaction and then you have to add this up. You'll get the correct answer here. So we'll write down. We have to find out enthalpy of formation of Ba2 plus, right? So we can write here Ba2 plus aqueous plus SO4. 2 minus aqueous, it gives BaSO4 aqueous, and for this delta H1 we can find out, which is this minus this plus this, and then we have BaSO4, it converts into BaSO4 aqueous. This is going to be delta H2. This is given, right, ma'am? This is minus of 4.5, right? Minus of 4.5 kilocalorie. Delta H1 is what here? Delta H1 is nothing but this plus minus this plus this, right? So we can write here minus of 349 minus this one, right? X plus sulfate minus of 216. So this is something, when you subtract, this is, let's say 1, I am assuming this is 2. When you subtract this 1 minus 2, you'll get it for this X. which is for BA plus 2. BA plus 2, plus 2. You'll get the answer from this one.
That is 120
Minus of 128.5.
that we can do by
This,
calculating these.
yes, this two we can add, we'll get the answer. Okay.
So the number of possible both the structure as well as the stereoisomers of cyclic ether. So I'm finding, sir, there's a language cyclic ether. Generally, student may done that mistake that they understood all type of ether, but they asked only the cyclic one. So let's go with the cyclic one only. So we have C4H8. So if I'm going with the 1, 2, 3, 4, and that is oxygen. 2 hydrogen, okay, everything is balanced. So this is the first possibility. Second possibility, I can make it like this way. 1, 2, 3, and 1. And we can make it one more pattern. That is the 3-member ring, we can make it. Okay, let me draw it properly. So like this, or we can make it like this way. One more possibility I'm finding, sir, there should be the ethyl group, right? So I think 1, 2, 3, that is 1, 2, 3, 4. Okay, everything is balanced. They asked about the structure in stereo. So there is no chiral, there is a chiral, and there is no chiral. There is these 2 chiral, and there is also chiral. So this is 2 to the power 1 is equal to 2. This is 1, this is 1. This is 2 to the power 2 minus 1 because, yes, mesomer is there. So we have 2, 3, and this is 2 to the power 1, 2. So total answer would be 2 plus 1, 3, 4, 5, 6, 7, or 8, 9. Yes. Let's check it. 1, 2, 3, 4, and this is 1, 2, 3, and 4. 1, 2, 3, 4. 1, 2, 3, 4. Okay, here everything seems fine. Some chiral is missing, is it? Let me check it.
The
Okay.
answer is 10.
Yeah,
Yeah,
no,
one is correct, we have
we missed, sir. This is so plane of symmetry is not there. That is the, so yes, that's what we missed it here.
So total we have 1 plus 2, 3, 4, 5, 8, 10 we have.
Now 10 is the right answer. Yes. Okay.
Okay, next question. Ma'am. We are
Yes.
just almost done. 4 more questions left, right, ma'am?
Hopefully, yes.
Yeah. Okay. So so we have to find out the correct order which is given here. Dipole moment is given, lone pairs on central atom it is given, bond length and bond enthalpy. Okay. So clearly you see here the last one that we have D. This is correct because we have a triple bond here, bond order 3, 2, and 1. So bond enthalpy, the correct. So D, so we have the correct obviously. Obviously, option 3 and 4 we can eliminate, right, ma'am?
Yes.
3 and 4 we can eliminate. Now we have to see either A or B, whether it is right or not. XeF4, we all know hybridization thing. We have 2 lone pair here. XeO3, how many lone pair? Obviously. XeF2, we have 3 lone pair. This we know all. So this is not going to know. So we can easily eliminate options in this. The correct answer is B is not correct. So A and D. H2O dipole moment, obviously it is more here than NH3 and then CH3. A and D, the correct. Right, ma'am?
So this is higher yield of NO. N2 plus O2 will give you the twice NO. Okay, Leach-Atelier principle there. And there is written plus. Plus means high temperature. So delta H value, if it is plus, means high temperature endothermic reaction. So we need a high temperature, lower temperature, wrong. Okay, Leach-Atelier is telling if you adding the reactant concentration, the rate of reaction will be higher and it move to the forward direction. So these two is also right. So we have A, C, and D. So that option is right.
So can we say in this, ma'am, if C is correct, D is also correct?
Yes.
So both should be there.
Yes,
Right?
right.
Okay.
So nitrogen and oxygen both is participating, so they both should be high.
Right. So A and first and second, so we can eliminate.
Yes, right.
Fine.
So means if you need to crack the competitive examination, you have to be smart enough to eliminate these answers. So KBC, you should
Yes.
know how to play. Okay.
Okay. Now the next is the rate constant of a reaction is given. How much time does it take to convert this concentration? Or do we have order mentioned here, man? Directly? No. We do not have, no? But this unit is given.
Right.
So we can predict from that the order of
And
the
we are teaching in the class that how you can identify the order if they are not mentioning whether it's a zero order or first order.
Right.
Yes, sir.
So what is the question? Question, how much time does it take to convert 7.2 to 0.9? Okay. Now, since I know this is first order reaction, so I'm going to have a, guess it like this, or you can use the expression for first order directly. Let's say after one half, it will convert into 3.6. Then one more half life if you go, it converts into 1.8. Then one more half life if you go, it converts into 0.9. This is what they're asking, 0.9.
Right.
That means how much time we are going to have? 1, 2, 3. 3 half life we are going to have. We have to find out 3 times t half, and that is the answer for this question. Rate constant is given t half formula, it is 3 times 0.693 by k, and k is 0.03. If you solve this, we'll get the answer. Is it right, ma'am?
Yes, sir. 69.3.
69.
Even those who doesn't know how to apply this short trick method, they can apply the formula directly and get the answer.
That's the same question we have discussed.
Yes, yes, yes. So I think in kinetics they have asked the 3 questions and quite similar question in the question they asked. So which one of the following reaction does not belongs to the Lasigney test? Okay, so we know that it is utilized for nitrogen, sulfur and halogen, phosphorus. So first equation certainly belongs to that sodium cyanide. Even this is also, this is the sodium extract, that is the Lasigney reagent. Even this is also right. Okay, this is not right. This is the quantitative analysis for the copper, for the carbon we were calculating. So the fourth is out of the race. So we will have answer is 4. So the answer should be the, yes, not belongs to 4.
Yes. So this is it, I think, for the discussion for the today.
Yes,
So
yes.
how do you think, ma'am? How was the paper like overall?
I'm finding this time physical chemistry, they have given pretty good weightage over the inorganic chemistry. Inorganic chemistry, as far as I'm seeing, only 2-3 questions. Organic also pretty less,
Organic.
but physical weightage is really high.
Was the paper lengthy, you think?
Yes. That those students who are unable to solve that question paper don't get disheartened because the question paper was really lengthy. So certainly, I believe every student must face the problem. So here the cutoff, I think, so it is gonna be down. Yes.
So we cannot expect 170 kind of marks.
Yes, yes. No. Because if you know the 100%, still I'm finding that is time consuming. Time consuming.
Even the options, that order and all, we have to see each and every option
Right.
properly.
And so for such type of question paper, you really need to be patient about that. That marks should not reach 720,
Right.
but still you will get the good marks.
This
over
was not the feedback that we received from the students and the other faculties.
Yes.
This paper was not for 700 marks.
Correct.
Exception students fine,
Huh.
but usually it is not 700.
Yes.
Let's see what happens. Okay, so this is it from our side. Thank you so much.
And now the physics teacher is coming, so just wait for the 2 minutes that physics teacher is joining you. Thank you. Thank you so much, sir.
Thank you, ma'am. Thank you, ma'am.
Okay, students, so physics teacher is joining us. I'm also waiting. Okay, Kalyan sir is there, so we can welcome him. Sir, now stage is yours, please.
Hello. Hello. Hey guys, so we had a look at the paper and we wanted to just have a general discussion regarding the paper as well. Me and Shivam sir both thought the paper was lengthy and tough. So I think attempting the paper according to its merit would be sufficient, and not thinking of a cutoff would have been a good approach to solving this paper, right? With that being said, shall we start solving the question? Okay. So there was this question as well. We both have tried for a solution for this question, but we think this is slightly out of syllabus as well. But according to what has been mentioned in many textbooks which we have gone through, and we have seen these formulas only in engineering textbooks and all. So with that in mind, the two formulas which have been mentioned over here is, one is delta S was equal to d2y by dx squared. The change in pressure, actually, the change in pressure was equal to S into d2y by dx squared. And the other equation which we can write for this is delta P is also equal to rho gy, and hence framing an equation from both of them, if we get rho gy is equal to S into d2y by dx squared. But to be fair, this is not a question we expect you to solve, nor where we be able to solve on our own as well, right? So this is not a question we're going to be discussing much. We'll be moving on to the next ones. Most probably this might be a bonus question is what we feel, right? Actually, I think let's share the mic.
Okay, so this question is not for NEET level, even for JEE. We study this in engineering physics. And this formula is called Laplace-Young formula, right? It's from engineering physics. So most probably this question is going to be bonus, right? We have never ever studied, or we never ever teach this kind of things in the class. So anyways, this is the Laplace-Young's equation, and this you already know, delta P is rho gy. We simply equated them and got the second option as answer, okay?
We are also very curious to know what the NEET people think of the solution, and we'll also like to have a look at their solution. But we'll move on to the next ones. If you had been very picky about choosing the right questions, we could have done well in this paper. For example, the second question is a pretty simple one. We just have to apply a direct formula for this question. Your magnification formula is L into D by F naught into Fe. And we get the answer for this to be equal to 125. Right? Moving on.
Okay. So in this next question, an electron is moving with some speed c by 100. It's entering a magnetic field this much, and we want the electron to pass as it is, right? If electron has to be passed as it is, it means it is a question of velocity filter. Now, velocity selector. So first, let's find out the velocity, what velocity the electron is going to have. So velocity is going to be E naught by B naught. So here, if we put the value, right, we are going to get this E naught, the electric field magnitude, is equals to velocity of what we want to calculate here is electric field we want to calculate, yes. So it will be V into B naught. V is given as speed of light, 3 into 10 raised to power 8. B naught is also given. From here we can get the velocity of electron, 27 into 10 raised to power 4, right? Now we know that in velocity selector, what is the angle between E and B? 90 degree. So in B are perpendicular to each other, and velocity is 27 into 10 raised to power 4. Hence first is the correct answer. Now moving on to the next question. So Kalyan sir is going to discuss it.
Okay, so as you could probably see, the previous two questions were slightly easier, right? So if you had gone about your questions properly and done this, you could have, if you had chosen your questions properly, you could have made a decent attempt in this paper. But this is also not a difficult question. The only idea in this question is, you have to do a bit of calculation. So in the first case, your object is moving down a rough inclined plane. In the second case, your object is moving down a smooth inclined plane. We need to find out the relation between their times. So the relation between the times is given as the time taken in the first case is twice of time taken in the second case. So direct equation we can write for this is root of, we can write half a t squared is equal to distance traveled. So time taken will be equal to under root of 2 distance divided by acceleration. And what's your acceleration in this case? Your acceleration in this case is g sine theta minus mu g cos theta. Whereas your acceleration in this case is nothing but just d sine theta only. So we just have to solve this equation, and when we solve this equation, we end up with the value for mu to be equal to 0.75. So this was a slightly lengthy question, but as you can see, the equation can be written directly, and then there's just a one-line question if you had to just solve the equation as well, right? Okay, then let's move to the next one. Okay, and this also is a pretty straightforward question. You had two cars having some initial kinetic energy. The first car had an initial kinetic energy of 100 joules. Second car has some another initial kinetic energy of 225. We are stopping them. We are stopping these two cars, and the force we are applying to stop them is making one car stop in a distance of 1000. And for the other car, the distance it travels before it comes to a rest is 1500 meters. So we need to calculate the ratio of the forces in this case, right? So here, can I say that work done is equal to nothing but change in kinetic energy? So we can say the force we are applying is removing the entire kinetic energy. So K1 by K2 will be equal to nothing but F1 S1 divided by F2 S2. We just have to solve this equation and then we end up with the answer is 2 by 3. Moving on. Circuit question. Okay. So in the circuit question, I think all you had to identify was this is a Wheatstone bridge. And once you identify this is a Wheatstone bridge, we can ignore the 6 ohm resistor. Then 3 and 5 are in series. 1.5 and 2.5 are in series. 1.5, 1.5, and then this 5.5 are all in series as well with this one. Right. So once you realize this is a Wheatstone bridge, these two are in series, that's 8. These two are in series, that's 4. 8 and 4 in parallel will give me 32 by 12, which is 8 by 3. Then we just add up all the net resistance. So we get the total current value, right? And actually, let me just calculate the R effective quickly. Your R effective here will be 8 by 3 plus 1 by 3 plus 5.5 plus 1.5. So your R effective is 10 ohms. So what will be your current value? Your current value will be 5 by 10, which is equal to 0.5 amperes. So this again was a simple question. There were difficult questions in this paper, no denying that, but you could have identified the simple questions and we could have gone ahead with it, right? If you have been smart about your questions. Right?
Okay, so another lengthy question is here, right? So here they are asking if V naught is velocity here and string is getting slacked here, what is the ratio of the velocities V naught and the velocity V here? Okay, so I will just write the equation and skip the calculation. We'll directly select the correct answer. So here, let's say the velocity of particle becomes V. Here it's V naught, and since if a string is getting slacked, it means tension in the string is 0. At this particular moment, we can say that the mg, gravity, and this is theta, this angle will be 90 minus theta. So component of mg cos 90 minus theta, or I can say mg sin theta, will be providing centripetal force. So I can write here that mg sin theta will be equals to mv square by r, or here instead of r they have given L. From here we can find the velocity at the point P, which is going to be simply root GL sin theta. Now how to find velocity V naught? For that we have to apply energy conservation between this bottommost and P point, that is half mv naught square will be is equals to energy at the P point half mv square plus gravitational potential energy mgl, that is this side L, plus component of L. So this component of L will be L sin theta. So mgl plus L sin theta. Now what you have to do is put the value of V here, do the calculation, you will find V. Now I am not doing it, but the ratio of V and V naught, like here we are going to find V naught. It is going to be option number 4, sin theta by 2 plus 3 sin theta square. Okay. Now moving on to the next question, and here we have this gate. We have to identify which gate it is. Okay, this is A, B and A. So here we are going to get A, B, B and A. It's OR gate, so and it's OR gate with a NOT, right? So NOR gate, so output will be A plus B whole bar. AND gate with a NOT gate, so A dot B whole bar. Now this is feeded into AND gate, so our final output Y is going to be A plus B whole bar dot A dot B whole bar. Now Boolean algebra we will apply, so A bar dot B bar dot A bar plus B bar. Now we can simply multiply this in the bracket, so A bar dot B dot A bar plus from here we are going to get A bar dot B dot B bar. Now see, we can say from here, okay, A bar B dot B bar, and here we are going to get A bar, I think here I have, so A bar dot B bar, yes. So here also we are going to get B bar, right? Here also. Okay, so what we are going to get, see here, I can write this as A dot A bar dot A bar. So this A bar dot A bar is simply A bar. And here I can say that B bar dot B bar is simply B bar. Now A bar dot B bar plus A bar dot B bar, it's simply A bar dot B bar. So which gate it is? It is NOR gate, right? So this is going to be when it's 0, when it's 1 and 1, then only the output is 0. So it is NAND gate. NAND gate means the option will be second one. Now, next one, I think Kalyan sir will help us with this one, right? Okay, I will solve this one. Next question, Kalyan sir will be solving it. Okay, electric field equation is given here, and we have to find the equation of magnetic field. It is given in the question itself that the equation of electric field is given, and the direction of wave can be calculated here. Since it's given in x, the electric field, the wave is traveling along x-axis. So wave is going along x-axis, electric field is along z-axis. So without a doubt, we can say that the speed of wave is E cross B, right? So from here, we can say that this is how we can identify the direction of the wave. So since this is z, this is x, it is going to be in y direction. So our answer can be this or this. Now how to find magnitude? Again, we will use that velocity selector or this formula, E naught by B naught is c. From here, we can, when we do the calculation, we are going to get the first option as our answer. So our answer is option number 1. Now, next question, Kalyan sir will help you with this one.
So this is a pretty straightforward question. You had an object being dropped from a height of 40 meters. The mass of the object is given, and then it's mentioned that this object is bouncing up to 10 meters, right? We need to calculate what's the impulse imparted to the ball. So the impulse imparted to the ball in this case is nothing but change in momentum. And the initial momentum your object has will be equal to m into root 2g of 40. And what's your final momentum? Your final momentum will be in the opposite direction. So it's minus of minus m of root 2g into 10. There's a simple equation which we need to use over here to solve this question. When you simplify this further, there's a pretty straightforward calculation only. We end up with the answer as 20 Newton second. Moving on. Okay, so this also is a pretty straightforward question actually. We just have to find out what's the potential difference between A and B. So let me write the equation. We'll write VA minus L into di by dt. We are going in the direction of current. Minus 5 minus I into 2 has to be equal to VB. And now it's just a matter of solving this equation. We'll get VA minus VB is equal to L into di by dt plus 5 plus 2I. L is 1, di by dt is 1, 5 plus 2 into 2. We get the answer is 10 volts. We'll get the answer is 10 volts. Moving on.
So in this question, this is actually, I think, this might be the simplest question of the exam. We have two circular loops and both of them have 2 ampere current. Their radius are different. We have to find the ratio of magnetic moment, that is mu A by mu B. Okay, we know that mu is nothing but I into A. So I A by, or I will write I1 into A1 by I2 into A2. But since current is same, we can cancel. I we can write as pi R1 square by pi R2 square. Now R1 by R2 square is the answer. Since R1 by R2 is 1 by 2, so its square will be 1 by 4. Okay, so this might be the simplest question of the exam, right? Now next one is this. We have 4 lenses, thin lenses placed close to each other. So what is the net power? We know that P equivalent will be sum of all the powers of P plus P plus P plus P, right? So it will be simply equals to 4P. But what is magnification? It's simply equivalent magnification will be m into m into m into m, right? So it will be m to the power 4. The correct answer for this question is p to the power 4p and m to the power 4. Now moving on to the next question. Here we have, okay, this one is a little bit lengthy question. We have to solve a lot. But anyways, I will just tell you the formula and what the answer is. So an oxygen cylinder has 30 liter volume. In meter cube, we can write it as 30 into 10 raised to the power minus 3 meter cube. Now, there are total 18.2 moles of oxygen in it. Some oxygen is withdrawn from it. And now, the atmospheric pressure has become the pressure of the container is 11 P0, 11 times atmospheric pressure, right? 11 into, we can take 1.01 into 10 raised to the power 5 Pascal. Okay, temperature they have given 27 degrees Celsius, so I can write it directly as 300 Kelvin. We have to find out how much oxygen we should remove from the container to achieve this situation. Now, see this. Here we have P, N, V, T, right? N is initial without withdrawing the oxygen. So let's say I have withdrawn some oxygen and final oxygen left in, final number of moles of oxygen left in it is N dash, right? So here if I apply PV is equals to N dash RT, I know P, I know V, I know R, I know T. From here I can find N, right? From our calculations, I remember it will come around like 13.2. N dash will be 13.2 moles from here. Now, so what is the number of moles left? So N left, the number of oxygen, sorry, the number of oxygen molecules removed. So removed oxygen will be the initial moles minus the moles which are left. So from here we are going to get around 5 moles. So if you want to know how much of the mass of oxygen left, it will be 5 moles. So mass will be, mass left, mass removed is going to be 5 moles, molar mass is 32, and to convert it to kg, you have to divide it by 1000. When we do that, we are going to get something approximately as 0.156 kg. So this is the answer of this question. Now, next one, Kalyan sir, to you.
So straightforward question, but the calculation is lengthy. It's a mathematical question. So here we are supposed to calculate what will be the acceleration, and we have a relation between t and x given. Rather than finding out x as a function of time, here if we just differentiate it directly, that would be a better approach. So what do we get from differentiating this? Is we get 1 is equal to 2x into dx by dt plus 1, or we can write 1 by 2x plus 1 will be your velocity. Since we need acceleration, we are going to be differentiating velocity once again, and that will give us minus 1 by 2x plus 1 whole squared into 2 into dx by dt, which is nothing but velocity. Now substituting back the velocity expression over here, and what was velocity expression equal to? 1 by 2x plus 1. So when I now substitute here, I'll get this as minus 2 by 2x plus 1 whole cube. Right? I think we had come across many questions of this kind in your module at least. So this is something which I would expect a student to solve, at least from our institute. Moving on. Okay, so here we have a AC power supply, 220 volt, and we have a resistor, a capacitor, and an inductor connected in series to it. We are supposed to calculate what will be your current in the circuit given the values of resistance is 20 ohms. All these values also have been mentioned in the ohm unit. So this will be 25 ohms and this will be 45 ohms. We are supposed to calculate what will be the current in the circuit and what will be the phase difference between the current and the voltage. So a normal phasor diagram will suffice. So what will be a phasor diagram over here? To find out the impedance, we can say that, or if I have to write exactly, the equation will be I into 45 for voltage, I into 20 for across resistor, and I into 25 for capacitor. On solving this, we get I into 20 root 2 has to be equal to your EMF, which is equal to 220. So we get your I value to be equal to 220 by 20 root 2, which is equal to 11 by root 2. So I think approximately it should be 7.8 amperes. And then what about a phase difference? Our phase difference formula is tan phi is equal to XL minus XC by R, which will become 45 minus 25 by 20. That's equal to 1. So phi will be equal to 45 degrees. A pretty straightforward question, a direct one. Let's move on. Okay.
So this question is again straightforward, right? So it is based on Kepler's law. So sun rotates around its, this is not based on Kepler's law, it is based on angular momentum conservation. So sun is rotating and it takes 27 days for sun to complete one round. Now they are saying hypothetically if sun expands to twice of its present radius, how much time it will take to complete one circle, right, or one full round. So we know that since we are not applying any torque from outside, the angular momentum will remain same initial and final. So I can say initial angular momentum, that is I initial omega initial, will be I final omega final. Now here the initial angular momentum will be 2 by 5 mr square. Initial angular velocity I am calling it omega or omega naught, yes. And here the final angular momentum will be 2 by 5. Mass will remain same, mass will not change. Radius has become twice, so I can write here 2r whole square and let's write it omega final. So here 2 by 5 m and m will cancel, r square will also cancel. So omega final will be 1 by 4 of omega initial. Since sun become 1 by 4 times slower, the time taken by the sun will be 4 times the original time it was taking. So new time, or we can say it like this, since omega is inversely proportional to time period. So if omega has become 1/4 time, the time period will be 4 times the original. So 4 into the original time, 27 days, which is going to be 108 days. Hence, the correct answer is option number 4. Now moving on to the next question. Kalyan sir, I think, will help us with this.
Okay, so this question was a tricky one. I think some questions could be, or some more conditions could be mentioned a bit more explicitly over here. But at least the first part of the question is clear, that is the flux passing through the electron. You have an electron moving in a circular path, and the flux passing through the electron is said to be quantized. Your flux passing through this electron is equal to NH by E. Right, so I'm just using the formula for flux. I'm saying that flux will be equal to magnetic field into area, so that's B into pi r squared, and that has to be equal to NH by E in this case. Okay, so what we are now supposed to calculate is what will be the magnetic moment of an electron in its lowest energy state level. Okay, so our magnetic moment formula, if you're familiar with, has to be equal to I into area, which in this case is I into pi r squared. Now instead of current, we'll be using E divided by 2 pi r by V, which will be EV by 2 pi r into pi r squared. So pi gets cancelled. So this is the term we are supposed to calculate. We are supposed to calculate this term EVR by 2, right? And we have one equation to help us get this. The other equation which we are using to help us get this answer is, we know that this is moving in a circular path because of magnetic field only. So your force, your magnetic force, which is EVB, has to be equal to mv squared by r. And now from this expression, we can make use of the r term, and from this expression we can make use of the velocity term. For example, what do I get my velocity here to be equal to? We get a velocity here to be equal to ERB by m. Now substituting this all over here. So this will be E by 2 into ERB by m into r by 2. So as you can see, this will become E squared B r squared by EVR by 2, V is ERB by m. I think I wrote a 2 twice. It will be r by 2m. So it's E squared B r squared by 2m. And now instead of r, substituting the value of what we got from this equation. What will r squared in this equation be? R squared in this equation is NH by E pi b. So substituting NH by E pi b. So E squared B by 2m into NH by E pi b. So one E gets cancelled. So we get the answer is, B also gets cancelled. So we get the answer is ENH by 2 pi. He's asking the energy of the electron at the lowest level possible. So we'll take the n value as 1. So the answer will be EH by 2 pi. So a slightly difficult question to figure out, but this is the way we need to be solving this question. So in my opinion, some additional conditions had to be mentioned, like this wasn't mentioned explicitly. Moving on. Okay. Now this is the direct question based on thermal conductivity. We'll have to accept steady state over here, and we'll be writing this equation, that heat transfer has to be same through both these junctions. So the equation I'll be writing is, we'll write 3T minus, sorry, I'll be writing heat flow equation. So my heat flow equation is 2K into A into 3T minus T1 divided by L has to be equal to K into A into T1 minus T2 divided by L, which again should be equal to 2K into A into T2 minus T divided by L. Right? So this looks like a difficult equation to solve, but if you're very clear about what you need to find out in this case, which is T1 by T2, should not take you too much of a time. It's a pretty straightforward question only. We'll get the answer as 5 by 3. Okay. Moving on.
Okay, so we are having another question on the capacitor. So we have the plates of parallel plate capacitors are separated by D. Now we are inserting two dielectrics of constant K1 and K2 such that K1 and K2 are related like this. K1 is 1.25 K2. The thickness of these dielectrics are 3D by 8 and D by 2. Now it is given that we have to find the value of K1, but it is given in the question that once we have inserted the slab, the capacitance becomes 2 times of the original capacitor. right? It becomes 2 times larger than the when there is no plate in between. So here they are saying the new capacitance C is 2 times the original capacitance C naught. Now here we can simplify the value of C naught to be epsilon naught A by D, and when the dielectric slabs are inserted, we have to apply the formula epsilon naught A by D minus T. So what is the thickness of first slab? 3 by 8 D. The second slab is D by 2, so minus D by 2 also. And finally we have to add D by this one, T by K, T by K. So first slab has thickness 3D by 8, K is K1, plus second slab has the thickness D by 2, right? And the dielectric constant is K. Now, what we have to do is, we are going to equate this to equation. The calculation is again going to be lengthy, but we are going to put the value of C here in this equation and the value of C naught in this equation. We are going to get one relation between K1 and K2. Another relation is given here, that K1 is 1.25 times K2. Now when we solve the two equation, one from here, one from here, we are going to get the value of both K1 and K2. And as I remember, the value of K1 is going to be 2.66. It will be nearly to 2.66. Okay, moving on to the next question. So here,
this is actually a pretty famous NCERT textbook question. It's something which everyone who solves NCERT textbooks would be solving. But anyways, let me go with the question. So you have two cities X and Y, where buses are leaving regularly from these towns every T minutes, right? So a girl is driving a scooty from X to Y maybe, and she's driving it with a speed of 60 kilometers per hour. So this person observes two things. The first thing that this person observes is, they observe a bus going past her. She observes a bus going past her, crossing her every 30 minutes in the direction in which she is moving. And then she also observes a bus crossing her in the opposite direction every 10 minutes, right? So we need to frame two equations based on these conditions. So let me first frame an equation for buses coming in the opposite direction. The first condition over here is to realize that the distance between two buses will be V into T, because let's say that your first bus is left this part. When will your second bus be leaving? Your second bus will be leaving after a time T minutes only. So during that time, your first bus would have already covered a distance V into T. So one idea is to realize over here that the distance between the two buses will be equal to V into T. So that's an idea I'm going to make use of over here, that the distance between two buses is V into T. And now let's say that the cyclist has seen the first bus crossing her. Now when will she see the next bus crossing her? The next bus will be crossing her after 10 minutes. And according to her frame of reference, the velocity this bus will be having will be V plus 60, because they're moving in opposite directions. So the equation I can write over here is, what does the time taken for the bus to reach this person? Is V plus 60 into 10. And what should that be equal to? So from this person's frame of reference, only this bus is traveling this entire distance. So V plus 60 into 10 has to be equal to V into T. Is the equation we'll write for the opposite direction. Now let me write a similar equation for the same direction. So let's say one bus has crossed her now in this. Where will the second bus be located? The second bus will be located VT distance behind. And now from this person's frame of reference, what velocity will this bus be having? The bus will be having a velocity of V minus 60. So the other equation we can write over here is V minus 60 into 30 minutes will be equal to V into T. So now it's just a matter of solving these two equations. And when we solve these two equations, we'll end up with the value of V and T to be equal to this. Moving on. So this was a lengthy question, but this is a question which is very standard. I think a lot of you must have done this during your NEET preparation. Okay. Now the next one, we have a uniform rod of mass 20 kg and length 5 meters. And now this is resting against a vertical wall, making an angle of 60 degrees with it. So this angle is 60 degrees, something to be very careful with. We need to calculate how much friction force is acting on this rod. So there are, let me draw the freeboard diagram. Let's say this is N1, N2, friction, mg are the forces acting over here. So one equation we can already write from this is N1 is equal to F, because this rod is in equilibrium along X. The other equation we can write is N2 has to be equal to mg. And now because it's in equilibrium, we can also write a torque equation. And this torque equation is preferably better to write about this point. So what will your torque equation turn out to be? Your torque due to mg will turn out to be mg into this distance, which is mg into L by 2 sine 60. And that has to be equal to N1 into this distance, which is N1 into L cos 60. And if you notice, your friction value is equal to N1. So from this equation, all we'll try to find out is what the value of N1 is. So L gets cancelled. We get mg by 2. Tan 60 is your value of N1, which in turn will become your value of friction. Mg is 20, so 200 by 2 is 100. Tan 60 is root 3. 100 root 3 will be your answer. Moving on. Okay. So in this case, we have an oscillating spring mass system. A spring is connected to a box filled with sand. And as this box is oscillating, it's found that sand leaks out slowly from this box. So we need to calculate a graph for how omega varies with time and how amplitude varies with time over here. And it's mentioned that this mass is leaking out of the box in, okay, nothing is mentioned about how this mass varies with time. Okay. Okay. So first of all, your omega for any kind of SHM in these kind of cases will be equal to K is equal to M omega squared. So omega will be equal to under root of K by M. And now because your mass is decreasing, and K is constant because K is dependent on the spring constant over here, and that is not changing in this case. So because M is decreasing, it's kind of obvious that omega has to increase. So one of your answers has to be omega has to be increasing over here. So we can already rule this out. Okay. Now to talk about the amplitude, how does your amplitude vary with time when mass of this oscillating system is changing over here? Since energy conservation should still be applicable in this case because there's no removal of energy from the system. So can I say half K A squared has to be constant? And because half K A squared has to be constant here, and your K value is a constant, so that implies A also has to be a constant in this case. So your graph will be A will not vary with time, whereas omega will be increasing with time in this case. So your answer for this would be your first option. Moving on.
Okay, so this question is directly based on dimension analysis, right? So here, instead of solving everything, we can just simply solve it from here. So this time is given as surface tension to the power alpha, then area to the power beta, then density rho, right? Rho is the density to the power gamma, and finally we have R. R is the radius to the power delta. Okay. If this equation is correct, we can compare the dimensions of both sides. So dimensions of time should be same as dimensions of surface tension. Surface tension is force by length. So MLT minus 2 by L will be MT minus 2 to the power alpha. Area is L square to the power beta. Density is ML minus 3 to the power gamma. And R is radius, then again length to the power delta. Now if we collect the powers, we are going to get m to the power alpha, then m to the power gamma. So m to the power alpha plus gamma from here, then t to the power minus, then we will collect L to the power 2 beta from here, minus 3 gamma from here, and plus delta from here. Now last, the time power, so time to the power minus 2 alpha, and that's it. Now just by comparing the LHS and RHS dimensions, here the time to the power is 1, there it's minus 2 alpha. So 1 will be minus 2 alpha, and hence alpha will be minus 1 by 2. Now if we check the options, we have this, two options where alpha is minus 1 by 2. Now I will use the m to the power is alpha plus gamma here, but on this side there is no m, so m to the power is 0. From here I can say that gamma will be minus alpha, and alpha is minus 1 by 2, so minus of minus 1 by 2 is plus 1 by 2. So the option which have alpha minus 1 by 2 and gamma plus 1 by 2 will be answer, which is only happening in option number 3. Hence 3 is correct. Now moving on to the next question. Here we have a question based on vernier calipers. So now let's see this. It is given that we have a vernier calipers where 10 vernier scale division is overlapping with 9 main scale division as per given in the question, and one main scale division is 0.1 centimeter. So 1 MSD is 0.1 centimeter. Now first I will find least count, then we will proceed. So to find least count, 1 VSD is going to be 9 by 10 MSD. This implies that the least count is 1 MSD minus 1 VSD, which will be 1 minus 9 by 10, that is 1 by 10. MSD. Now 1 MSD is 0.1, so 0.1 by 10, which will turn out to be 0.01 centimeter. Okay, so first part we have found out least count of vernier calipers. Now we can proceed and find out the measurement. It is given that when the jaws are touching each other, the vernier calipers is already reading and it's reading 0.1 centimeter on the main scale. So if main scale reading is 0.1, we can say that the zero error will be 0.1 centimeter. Now, next part of the question, the diameter is measured and M, the main scale reading is 5, vernier scale reading is 8. So now we can say that the correct reading is main scale reading plus least count into vernier scale reading minus zero error. Main scale reading is 5 centimeter, least count is 0.01 cm into the vernier scale reading, which is given here. Main scale is 5, vernier scale is 8 minus 0.1. Now here 5 minus 0.1 will be 4.9. 4.9 plus 0.08 will be 4.98 centimeter, which looks like option number 3 in this case. Okay, next one we have is a theoretical question, and I think Kalyan sir will help us with this one.
Okay, so in this question, we have a parallel plate capacitor which is being charged. So the question being asked over here is how will the magnetic field vary due to the displacement current, right? So first we will try to calculate what the displacement current will be equal to, and if you're familiar for the expression for displacement current, that's epsilon naught into d phi by dt. So it'd be epsilon naught into d by dt of sigma by epsilon naught, which is your electric field value, multiplied with the area of the capacitor. So epsilon naught gets cancelled. So what we end up with over here is we get the expression as A into d sigma by dt. And here if you notice, it's mentioned that this is a constant. So the condition we get over here is displacement current is a constant over here. But the question is not about displacement current, the question is about how the magnetic field varies due to this displacement current. So now how can I imagine this? We can imagine this actually as a long cylindrical wire having uniform current density everywhere because field is uniform everywhere. And now how will your magnetic field to a long cylindrical wire be? It will be increasing from center to the surface, and then after the surface it is inversely proportional to R. It will be decreasing, right? So you'll have magnetic field at all the places. You'll not only have magnetic field in between the plates, and your magnetic field will be maximum at the surface of the cylinder. So I think the third option is correct. The magnetic field is non-zero everywhere with maximum at the imaginary cylindrical surface is correct. Moving on.
Okay, again one very simple question, right? So an unpolarized light is falling on a surface at a Brewster's angle. We have to find out which option is correct. Now when light ray falls on Brewster angle, the reflected ray is completely polarized. Only option which have this is this one. The reflected light is completely polarized. So without even looking anything else, this is the answer. Because in other option you can check reflected light is partially polarized. No. Both reflected and transmitted light are perfectly polarized. No, because the transmitted ray is partially polarized and reflected is completely polarized. Now last one, transmitted ray is completely polarized. Again incorrect. So no need to calculate angle or anything, just theory can help us to solve this one. Now the next one, I think it's yours.
So we have two identical charge conducting spheres. The charge on each sphere is E. We need to calculate what will be the force acting between them according to certain conditions. Okay. So initially both these spheres had the same charge. They had a charge Q. They were separated by a certain distance. So the force expression in the first case will be equal to a simple formula KQ squared by R squared. Now it's mentioned that we are bringing a third identical sphere, and this third identical sphere was uncharged. It was initially kept in contact with A. So when I keep this in contact with A, the charge on this becomes Q by 2 and the charge on this becomes Q by 2. And now when I keep it in contact with C, again the charge splits equally between them. So now the final charges you will have on both the spheres is you'll have Q by 2 on this, but you'll be having Q plus Q by 2 by 2, which will be equal to 3Q by 4. So what will be a new force acting between these new spheres then? The new force acting between these two spheres will be K into P by 8 of Q squared by R squared, which is nothing but 3 by 8 times of your old force. So it will be 3F by 8. Moving on. Okay, so this is also one more question which I felt could have done with some more information. So you will see me assuming some assumptions over here to get the answer. And that is, you have two containers having chambers of volume. So we have two containers given. All information regarding the gas kept in these containers are given. N1 is 5 moles here. V1 is 2 liters over here. And the initial pressure of this is 1 atmosphere. Similarly, here we have N2 is equal to 4 moles. V2 is equal to 3 liters and P2 is equal to 2 atmospheres, right? And there was some sort of partition between them, a thermal partition which has now been removed. So you need to calculate what final pressure this mixture will be attaining over here, right? So generally when people look at this question, they try to apply conservation of mass directly, but here the idea we need to proceed with forward is conservation of energy. And because nothing is mentioned about what kind of gas this is, I'm assuming that both the gas are having the same degrees of freedom. So we'll assume that both the gas have degrees of freedom F. And now I'll just be writing a conservation of energy equation, which is nothing but N1 F R T1 by 2 plus N2 F R T2 by 2 has to be equal to N1 plus N2 into F R final temperature, which can be different, divided by 2. So this is the equation we'll be trying to solve right now, but as you notice, there is nothing mentioned about temperature over here. So that is one thing we'll have to contend with. So anyways, solving all the common parts. And now we have an equation N1 T1 plus N2 T2 has to be equal to N1 plus N2 into final temperature. So since temperature is unknown, we'll now try to write this in terms of pressure and volume. So my equation will become T1 V1 by R plus V2 V2 by R is equal to final pressure into final volume divided by R. And now R gets cancelled in all these equations. So now we need to solve this expression, and what will our answer turn out to be? So P1 V1 is 2 into 1, so 2. P2 V2 is 3 into 2, so that will be 6 divided by the entire volume. And what will be your entire volume in this case? That will be equal to 5. So you get a new pressure, and the new pressure value will be 8.5, 8 by 5, which is 1.6. Okay, moving on. Okay. A particle of mass m is moving around the constant force. Okay, so if you have to now imagine that instead of the centripetal force, which is generally the electrostatic force of attraction, if you had a constant force making this particle move in a circular path, then what would your formula of Bohr's orbit become? So we'll once again go with the derivation, and the two equations we'll be writing for deriving all your expressions is mvr is equal to nh by 2 pi and mv squared by r is equal to, in this case, instead of writing kq squared by r squared, we'll be using the expression f. Okay, so we'll go with a normal derivation method, which is we'll find out v is nh by 2 pi mr. Substitute that expression over here. What will I get? I get m by r into n squared h squared by 4 pi squared. You need not focus on the constants much. 4 pi squared m squared r squared is equal to f. Okay, so now all I need to focus on is r and n term. So how is r varying with n is our main focus over here. So here we get r cube is proportional to n squared. So from this we can say that r will be proportional to n power 2 by 3. R will be proportional to n power 2 by 3, right? Okay. Now the next part, how do I find out the velocity expression? So once again, for velocity expression, we'll say v is equal to nh by 2 pi mr. And again, not focusing on the other terms, only on n and r. So it's n divided by r, which was proportional to n power 2 by 3. So we'll get velocity proportional to n power 1 by 3. So our answer will be the third option. Moving on.
Okay. Now in this question, the radius of Martian orbit, Mars is revolving around the Sun, and its radius is 4 times the radius of the Mercury. The Mars is taking, for 1 year on Mars is 687 Earth days. Then how much is the length of 1 year on 1 year Mercury on Earth? How much days it will be? Now see this. This is the question of Kepler's law. So we know that for Kepler's law, T square is directly proportional to R cube. So we can say that T1 by T2 will be R1 by R2, and I am taking the power as 3 by 2. According to question, the time of first planet is given as 687 Earth days. Second, no idea how much it is. R1, the radius of first planet, that is Mars, is 4 times the radius of Mercury. So I can write it like this. R and R is cancelled. We are going to get 4 square root is 2, 2 cube is 8. So 687 by T2 will be 8. From here, the T2 will be 687 by 8. If you solve it, it will come around to be 8 8s are 64, 47. If we make approximation, 8 5s are 40, 8 6 are 48. So nearly 86 days. So this question, the correct answer is close to 88. Hence we can choose 88 Earth days. But maybe since answer is not exactly matching, there is a good chance it might also become a bonus question. So who knows? We have to wait for the official answer key. Okay. Next question is a body weighs 48 Newton on the surface of Earth. We are taking it to a height which is equals to 1/3 of the Earth's radius. What is the new weight of the object? Now see this. Original weight is nothing but the force applied by Earth, which is Gmm by R square. According to question, this is 48 Newton. At a distance of, or at a height of 1/3 the radius of Earth, the new distance from the center of Earth will be R plus R by 3 whole square. So new weight will be Gmm by R plus R by 3 whole square. Taking LCM, it will be Gmm, 4 in denominator, 3 in numerator, that is squared. So 9 by 16 times R square. I can put Gmm by R square as 48. So 9 by 16 into 48. So 3 9s are 27 Newton. Now 27 Newton is the answer. This is also one of the easiest question of this exam. Now moving on to next one. A wire of resistance R is cut into 8 equal parts. So resistance of one part is going to be by 8. Now what they are saying is, out of this 8 pieces, 4 are connected in parallel, rest 4 are also connected in parallel. And this parallel and parallel are connected in series. What is the equivalent resistance in final case? So when 4 are connected in parallel, the resistance of this one combination, or this 4 are connected in parallel, right? So resistance of one parallel combination is going to be resistance of 1 by 4. So it is going to be R by 32. Now two such resistance are connected in series. So final R equivalent will be 2 into R parallel. So 2 into R by 32, which will be R by 60. So correct answer is third one. Another easy question. Now next one, Kalyan sir will help us.
Okay, so if you go with the calculations in this question, I think we might not end up with the exact answer. But let me go with it anyways. We need to find out the de Broglie's wavelength of an electron revolving in the second orbit. So the formula we'll have to use over here is h by mv. But we know mvr has to be equal to nh by 2 pi for Bohr's orbit. And now the n value I have to substitute over here is 2. So I know that mvr here will be equal to h by pi. I don't need the entire mvr, I just need mv. So mv will be equal to h by pi r. And now we need to substitute this in this formula. So this become equal to h by pi r. But now because you're finding out the r value of the second orbit, so the r value I need to be substituting over here is 4 times of, it's n squared by z, so it's 4 times of 0.052 nanometer. So this will be equal to h divided by pi of 4 into 0.0.2. So this will give me somewhere near 1.67 divided by 4, right? And there's no such option over here in this case, right? But this will be the answer for this question. It will be h by pi divided by 4 into 0.052. Moving on. Okay, so this is also a pretty straightforward question. We need to calculate what the change in potential energy. So the formula is pretty direct. It's minus delta U is equal to minus P cos theta final plus P cos theta initial. Your theta initial value is 0 degrees. Your theta final value will be equal to 60 degrees. Substituting this will be minus PE by 2 plus PE, which is equal to PE by 2. Both the values of P and E are given. This is 5 into 10 power minus 6. This is 4 into 10 power 5 divided by 2. So 22 1 joule will be your answer. So again, one more straightforward question. There were a few of these in this paper.
Okay. Now in this question, we have to find out the current flowing through CD. First, we'll check if it is a Wheatstone bridge or not. So 1 and 3, 2 and 4, it is not. Now, what I will do is, these two are in parallel. These two are also in parallel. So I will find equivalent resistance of this portion and this portion. Now, so equivalent resistance of this is 1 into 3 by 1 plus 3, that is 3 by 4 ohms. So this whole portion has a resistance of 3 by 4 ohms. What about this? The equivalent resistance will be 2 into 4 by 2 plus 4, 8 by 6, 4 by 3. So this much portion have a resistance of 4 by 3 ohms. Now this whole circuit is going to have the resistance of R equivalent is equals to 3 by 4 plus 4 by 3, 9 plus 16, 25 by 12. Hence, the current sent by the battery will be V by R. E by R equivalent, E is 50 by 25 by 12, 24 amperes. So, how much current the battery is sending here? 24 amperes. Now, see this. The equivalent resistance of this part is 3 by 4. So, hence the voltage drop V1 across this whole circuit or this much portion of the circuit will be IR, right? Let's call it R1. This is R2. So, IR. So, across this, the potential drop will be 24 into 3 by 4, 18 ampere. So, it means current flowing voltage, 18 volt. Okay, so here the voltage drop is 18 volt. And if this is 18, the remaining potential across this will be 50 minus 18. And 50 minus 18 is going to be 32 volt. Okay, if 18 volt is here, then current flowing across this resistance will be I will be V by R. So current flowing I1 is going to be 18 by 1, that is 18 ampere. Now, what about current flowing through 2 ohm resistance? The voltage across this whole part is 32. Current will be V by R. So I2 is going to be 32 by 2, 16 ohms, 16 ampere. So hence, we can say that 16 ampere current will go here. So out of 18, 16 is going there. How much should go there? 2 ampere. So current will flow from C to D. And how much the current will be? Just 2 ampere. So hence the correct answer for this question is going to be option number 2. Now moving on to next question, here we have to find the ratio of the wavelength of photon and electron. It is given that they have same energy. So energy of photon and energy of electron are same. Photon have energy h mu, which I can write it as Sc by lambda photon, and here the energy of electron, I can say that it will be half mv square, but we want wavelength, that's why I will write energy in terms of momentum, p square by 2. From here I can say that let me write it both them as E. So from here the momentum of electron is going to be root 2 m. So what is the wavelength of electron? The wavelength of electron is going to be h by p. So h by root 2 me. Now what is the wavelength of photon? Since Sc by lambda p is E, lambda p will be Sc by E. I will just simply derive, since we want lambda p by lambda E, we'll derive these two equations in reverse. So when we divide it, Sc by E by this thing will become reverse, h root 2 m E, h is 1. This will cancel and make it root E. So our answer will be C into root 2 m by E. It's 2m root 2m E by C. Third option is correct. Now next question, Kalyan sir.
Okay, so this will be a direct question once again. There's a relation of how photoelectric current varies with frequency and how photoelectric current varies with intensity as well. Right, so as intensity of light increases, the number of photons striking the metal surface also increases. And so as number of electrons released increases, we know I is proportional, right? So this graph is obviously correct. This is a pretty common graph. and this graph will be correct over here. So the relation between photoelectric current and intensity of light is correct. Now one more option which you need to check over here is what's the relation of photoelectric current with frequency of light. Now it is one point of discussion that as frequency increases, your maximum kinetic energy could be increasing, so that could also be an increase in the current value. But then we know that the relation which current shares with drift velocity is I is equal to NEAVD. So it's not proportional to velocity, it's proportional to, like, we get half mv squared is equal to h mu minus phi. So here if you see the relation which velocity has with frequency is not a directly proportional relation, it's more of a curved relation. So we'll definitely not have a straight line in this equation. So with that kept in mind, I would say D is not the answer, or either of C. Only A has to be the answer then for this question. Moving on.
Okay, in this question, we have a sphere from which a cavity is removed. We have to find out the ratio of moment of inertia of this removed part, smaller sphere and bigger sphere. You have to find ratio of their moment of inertia. For small sphere, let's say mass of small sphere is m, right? So this removed portion, its moment of inertia will be 2 by 5 mr square. What about the complete sphere? Its moment of inertia will be 2 by 5. Since mass is 4 by 3 pi r cube, mass is volume into density. So mass will be 4 by 3 pi r cube into density. Mass is directly proportional to r cube. Since the bigger sphere has radius twice, its mass will be 8 times the mass of small sphere. So the moment of inertia of complete sphere is 2 by 5 m r square. R is 2r. This square we are going to get 2 by 5 into 4 8s are 32 mr square. Now we want the moment of inertia of this bigger sphere with removed portion, which I am calling it as IB. So IB, the bigger sphere, I can say that the moment of inertia of bigger sphere plus moment of inertia of smaller one will be moment of inertia of complete sphere. So bigger one will be complete, which is 2 by 5 into 32 mr square, minus the smaller one, which is simply 2 by 5 mr square. 32 minus 1, here we are going to get 32 minus 1, right? So it is going to be 2 by 5 into 31 mr square. So let me just check the calculation once. So now this is the moment of inertia of smaller sphere about its own center. In question they are asking the moment of inertia of smaller sphere about the center of the sphere. So moment of inertia of the smaller sphere about the center, it's mr square by parallel axis theorem. We know this 2 by 5 mr square. So this much will be mr square plus the moment of inertia of small sphere about its own center. So now it is going to be 7 mr square by 5. They are asking the ratio of this bigger one and small about the center of the original sphere. So I big by small. So we are going to get 2 by 5. So big by small, we are going to get 2 by 5 into 31 mr square by 7 by 5 r square. Mr square will be gone. 5 and 5 will also be gone. We are going to get here 62, right? So we are going to get 62 by 7. So I think I have done some calculation mistake here. Somewhere radius 2r. Where 2r? R. 2r, right? So this 2 for the 32. So 2 by 5 into 32 minus 2 by 5. Here is the mistake. Here I have to substitute the moment of inertia of this small sphere, right? Small, which I have corrected here, is 7 by 5 mr square. Hence it is going to be how much? 2 by 5 into, so here when we are going to solve it, 64 minus 7, it is going to be 57 mr square. Now, you have to take the ratio of bigger and small. So now let's do it again. So bigger by small, it is going to be 57 by 7. Right? They are asking the reverse ratio. So hence the option will be third answer, third option. Okay, next part. So Kalyan sir, again, the diode question is here.
So this is a question based on a full wave rectifier, right? And we have an AC source connected across a full wave rectifier. Now, one thing which could have been mentioned in the question is there's no sign of what could be the positive polarity, what could be the negative polarity over here. That I think is an information which could be mentioned in this question. So here I'm going forward with the assumption that when you're in the positive half cycle, we have plus here and minus here. That this is at a higher potential with respect to this part. With that kept in mind, let me solve this question. So the time of 15 milliseconds, what will your potential difference across, what will your EMF value be equal to? That will be 220 sine of 100 pi t, and t is 15 milliseconds, so that's 15 by 1000. So this value turns out to be 220 sine of 3 pi by 2, which is equal to nothing but sine of pi plus 3 pi will be a negative value, it will be minus 220 volt. So basically this tells me that this is in this negative half cycle, and if it's in its negative half cycle, then that means this is now your positive part, this is now your negative part. Again, I think there's an information which could be mentioned in this question because we have no idea when this is in positive polarity, when this is in negative polarity. But if this is in positive polarity and this is in negative polarity, then that means that even here it will be positive and here it will be negative, in which case this diode will be in forward bias and this diode will be in reverse bias. So with that kept in mind, then I would say that the answer would be D2 is forward bias and D1 is reverse bias. Moving on.
Okay, so here the question is, two gases A and B are filled at same pressure in separate cylinder with movable piston, and RA and RB are the radius of these two pistons. On supplying an equal amount of heat to both the systems reversibly at constant pressure. So pressure is constant, that's the important point. A and B are displaced by 16 centimeter and 9 centimeter respectively. If change in internal energy is also same, we have to find RA by RB. So see this, same heat supplied, same change in internal energy, and pressure is constant. So if I use the first law of thermodynamics, that is del Q is equals to del U plus W. If this is same, work done will also be same. Work done has to be same. So I can say that W1 will be W2. Since it's constant pressure, the work done will be P delta V, or I can say P1 or yes, P1 delta V, and here delta V1, P2 delta V2. Now, it is also given the pressure is same, so hence we can cancel the pressures delta P and delta P. So delta V1 is simply same as delta V2. Now, what is the volume of cylinder? So pi R1 square, right? And let's say it has moved by a distance of d. So pi R1 square into d1. And here the volume that we are going to get is pi R2 square into d2. Pi and pi is cancelled. So hence the ratio of R1 and R2, it will be d2 by d1. The distance moved to the power half. The distance moved is 16 and 9. So their reverse ratio will be 9 by 16. We are taking a square root, so it will be 3 by 4. So hence the correct answer to this question will be 3 by 4. The RA by RB will be 3 by 4. Okay, next one. This is again very easy question. P is given as this equation. Error in all the quantities are given. We have to find the percentage error in P. So percentage error in P will be 3 into percentage error in A plus 2 into percentage error in B plus 1 into percentage error in C, and since d to the power half, so half into percentage error in D. Now A, B, C, D, all percentage errors are given. When we put this value and do the calculation, we are going to get the percentage error as 13%. So after putting the values, the answer will be third option, 13%. Okay.
This also is a pretty standard question based on Polaroids. So you have two cross Polaroids in this case. So let me draw those two Polaroids. Let me assume this is the pass axis of the first Polaroid, and this is your pass axis of the last Polaroid, which is, let me assume that's the third Polaroid. Your second Polaroid is slanted at an angle of 22.5 degrees with one of the Polaroids over here. So what we are supposed to calculate is what will be the resultant intensity after the light passes through all the three Polaroids. So if I go one by one, if you have unpolarized light passing through the first polarizer, because it's unpolarized light, the intensity of light which comes out here will be equal to I naught by 2. And now this will be polarized along the path axis of the first polarizer. So my first light will be inclined along, will be polarized in this manner. And now it makes what angle with the path axis of the second polarizer? It makes an angle of, let's say, theta for now. So what will be the intensity of light coming out from the second polarizer? The intensity of light coming out from the second polarizer then will be equal to I naught by 2 cos square theta. And now the light which is coming out from the second polarizer will be polarized this way. So then it makes an angle of 90 minus theta with the third polarizer. So the light which comes out of the third polarizer will have an intensity of, so let me write the resultant intensity. It will be I naught by 2 cos square theta into sine square theta. And since sine of 22.5 is difficult to calculate over here, one thing which you could do is you could multiply and divide it by 4. So this becomes I naught by 8 into 4 cos square theta sine square theta, which can be written as 2 cos theta sine theta whole square. So it becomes sine of 2 theta whole square. So this becomes I naught by 8 sine 45 whole square. Sine 45 whole square is 1 by 2. So you get the answer is I naught by 60.
Okay. Okay. In this question, it is given that two identical point masses P and Q, identical point masses suspended by two massless spring having spring constant K1 and K2, and they are doing SHMs. The maximum speeds of these two SHMs are same, and we have to find out the ratio of their amplitudes AQ by AP. So see this, if their maximum speeds are same, V1 max is same as V2 max. What is the value of V1 and V2 max? Omega 1 A1, and here it will be omega 2 A2. Now here I can write omega as here we know that for a spring system force is minus kx. We can write this force as ma, and a is going to be minus k by m into x. Comparing it with the standard equation of SHM minus omega square x, I can say omega for spring block system is simply equals to root k by m. Now putting the value of omega back here, we are going to get root k1 by m into a1 will be root k2 by m into a2. M and m is gone, so hence the ratio q by p, second by first. So a2 by a1 is going to be root k1 by k2, which is option number 4th in this question. Now next one, Kalyan sir.
Okay, so this is also a pretty standard question. In the first case, we have a organ pipe which is open at both ends. So what would be a fundamental frequency for an organ pipe which is open at both ends? The fundamental frequency is given as f in this case, and that would be equal to v by 2. In the next case, what we are doing is now we are dipping this in water. So now this becomes a closed organ pipe having a length of L by 2. And what would be a fundamental frequency in a closed organ pipe having length of L by 2? The formula will be V by 4L. So in this case, it's V by 4 of, but a new length is L by 2 in this case. So new frequency also turns out to be equal to V by 2L, which is equal to nothing but your actual fundamental frequency. So this is a pretty straightforward question. So the answer will be equal to. Okay, so this concludes our paper discussion. So one point which we noticed is when we did solve the paper, we did find it difficult. We did find it a bit lengthy, even though we have finished this discussion in a certain amount of time. But on introspect, the question seems a bit easier. But during the exam time, obviously the paper would seem difficult. So I would refrain you from jumping to any conclusions regarding, what the cutoff of the paper might be and all. You've given your best. So stick to it. And I think the cutoff will be a bit low this year, looking from what the paper is. Right?
Yes, it will be even in physics, chemistry, biology, everyone, all the topics, all the chapters were, all the subjects were tough only. So we are thinking cutoff will fall by a huge amount this time. So even if your paper has not went good, you have left a lot of question in physics. No need to worry. It's the same paper for everyone. Everyone is going to leave same amount of question. All of them are going to score almost same marks only. So the cutoff will fall by a lot of marks, but don't worry, it will fall for everyone. So even if your physics paper doesn't went that well, it's fine. After some time, once you are going to get answer key, just check it. So it's no need to worry. Okay. So that's it. It concludes our paper discussion. I hope every one of you has scored good marks. So best of luck, and we'll see you again next time. Okay. Okay, sir.